the quality of water, like to compare the tailing contamination of water in two study areas, maybe with respect to the health of the goats in those two areas. Thank you.
I didn't get your question properly. If you're asking whether I conducted the water samples, yes, I did on this slide.
A heterogeneity test to compare has more contaminated water with respect to the health of the goats.
You're saying, did I conduct the heredity test?
Heterogeneity test.
No. No, I didn't. I only conducted water samples from contaminated and uncontaminated areas, and then these are the results that I got. I didn't conduct that.
Thanks, Dr. Srakala. This is a good work. Just clarify me, what is the threshold for each of the chromium, lead, copper, and others that you said they were high? By how much, or what is the threshold, and by how much has your result exceeded that? That's one. Quick one. Two is that, how did you determine the health status of the goats? Was it through questionnaire?
Yes.
I think you may even get better results if you were to take some of the samples. Let's say maybe you buy a goat or whatever the number that you may have, and take some samples to the lab to validate what response you got through questionnaires. And I'm wondering whether if dust is the only issue, as you said, or there is also water spillage that it's also a problem.
Can I answer? Oh, yeah. We again. This is the first chapter, and then this is chapter 3. On chapter 4, we then conducted blood samples. We collected blood, and then of goats between contaminated and contaminated area, more than the threshold. Also with magnesium and lead. By how much? I can do the calculations now. It's fine.
Study and the previous studies. So like, what gap is your study filling just to compare to the previous ones?
Not much has been covered on the effect of tillings with regards to goat health. Previous literature has only covered the aquatic life, which are fish contamination to fish health, as in the aquatic industry, and then not taking into consideration the effect on goats' health. And then that's where my study comes in, to raise awareness that not only the aquatic industry is affected, but also livestock.
Thank you so much, Mr. Marakalla. Can we give him a round of applause? Our next speaker is Makwati. Makwati, are you with us? The stage is yours.
Good morning. I am Papom Makwati. I'll be taking you through the study titled The Determinants of Climate Change Perception and Adaptation Among Small-Scale Farmers in Limpopo Province and Application of the on Environmental, Social, and Economic Effects. Thus, it is imperative to understand how small-scale farmers perceive and adapt to climate change as a crucial step in guiding future adaptation strategies, deemed to be essential for the successful adaptation in agricultural sector. This study identifies several challenges that are low technological and financial capacities. Furthermore, the extent to adapt to climate change is poorly influenced small-scale farmers' perception and adaptation to climate change in the Limpopo province. By addressing this knowledge gap, policymakers and stakeholders can gain valuable insights on how to support and enhance climate change adaptation strategies. This research could inform the development of effective policies and interventions that can help mitigate the impact of climate change in the agricultural sector within this province. The aim of the study was to profile the socioeconomic characteristics of small-scale farmers, analyzing factors influencing their perception and adaptation towards climate change in the district of the Limpopo province. The specific objectives of the study are to profile the research hypothesis stated that socioeconomic factors do not influence small-scale farmers' perception and adaptation towards climate change in the study area. For the research methodology, Waterberg and Skukune district, several small-scale farmers, crop and livestock farmers identified within the sample size. On this table, it indicates the total number that the researcher accessed, which is 2,619 study. Descriptive results and discussions. For empirical data analysis, this study employed the Heckman-Probert model to analyze the perception and adaptation to climate change by small-scale farmers within the district. Climate change is identified as a two-stage process involving perception and adaptation. For the multinomial model, this provides a direction in which the effect of the independent variables model to the dependent variable. The explanatory variables were chosen based on previous studies. The first stage of the Heckman two-stage equation model is the perception to changes in the climate change and is used as the selection model, while the second stage is whether the small-scale farmer adapted to climate change. Conditional on the first stage, the farmer perceived that there is change in the climate. This table indicates the description model of the variables which have an outcome on the impact of. Figure 2 indicates the perception of temperature for the number of hot days during the summer, which was sampled from the small-scale farmers which were chosen for the study, whereby 72% of the small-scale farmers indicated that there is an increase in the number of hot days during the summer season, whereas 6 small-scale farmers were uncertain. Figure 3 indicates the perception of rainfall for the number of rainy days during the rainy season, whereby 74% of our small-scale farmers indicated that there is a decrease in the rainfall during the rainy season, and 4% indicated that the level of rainfall stays the same. For empirical data analysis, the Heckman two-stage model result of the sample small-scale farmers within the district is indicated on the table, whereby we identified that we have variables that are statistically significant to the study. For example, age is a statistically significant variable in this study, which means that it has a statistical significance to the dependent variable. For conclusion and policy recommendation, the null hypothesis of the study was rejected because the result of the Heckman two-stage equation model showed that age, education level, farming experience, number of adult laborers, experience crop failure, livestock loss, access to credit, access to extension services, farm size, perceived changes in the temperature and rainfall had a significant effect on the perception and adaptation towards climate change. It is thus recommended that the South African government should ensure that future policies focus on the awareness of creation and the campaigning on climate change through different sources such as mass media and extension services. Those were the references that were used to compile this presentation. I would like to acknowledge my supervisor, Dr. Nkwana. Thank you.
Thank you so much, Ms. Makwati. Can I have 3 questions for her? The first one over there.
Thank you very much, Makwana, for your nice presentation. My question is, what are your plans for future studies related to this research?
The future plan for this, from this research is to, the research mainly focuses on providing strategies that small-scale farmers can adopt, and this can help with policy making and decision making. So for the future researchers, they can make future studies on how this adaptive strategies can be further be improved for small-scale farmers and how they can be adopted.
Any other questions for her? Okay, thank you so much, Ms. Makwati. Can we give her a round of applause? Nkosi, are you with us now? Nkosi, Sinhle? Okay, then our next presenter is Bopape, Paul Bopape. Paul Bopape. Unfortunately, Bopape will not be able to present, so can I call Tloyamma? Tloyam. Tloyamma, are you with us? Okay.
Greetings, everyone. I go by the name of Mr. Tluwama TM. I'm here to present a study titled Distribution of Particulate Organic Matter in Bulk Soil from Areas Undergoing the Expansion of Banana Production in the northern part of the Kruger National Park, Limpopo, South Africa, under the supervision of Mr. V. Imbanjo. The particulate organic matter is a fraction of the total organic matter which does not pass through a filter pore size that ranges in size from 0.053 to 2 millimeters. POM accounts for 20% or more of soil carbon depending upon the agro-ecosystems and the management practices. The POM also improves the aggregate stability, facilitates water infiltration, increases the cation exchange capacity, helps buffer the pH levels and binds environmental pollutants such as heavy metals and the pesticides. Inherent and dynamic soil properties and human activities that affect the biomass production, accumulation and or decomposition will affect the POM content in the soils. So as agriculture continues to encroach upon the natural ecosystems, it is imperative to assess these changes affecting the POM content of the affected soils. The problem statement: the Baobab fruit farm has converted more than 400 hectares of the native grassland into banana fields over the years. However, they do not have the records of the impacts of these changes on the soil's physiochemical properties. So studying the physiochemical properties after the conversion is essential for sustainable land management, optimizing agricultural productivity, and minimizing the negative environmental impacts. So this study was thus initiated to assess the changes in the soil health, quality, and functionality following the conversion of the native grasslands to arable agriculture by quantifying the particulate organic matter. So this is a picture of how the native grassland and the banana fields looks like. The motivation of this study was to understand the land use change effects on the particulate organic matter and other properties across the farm, which provides the necessary data for informed decision making and helps to ensure the long-term viability of agricultural activities on the converted land. And the POM content would also indicate the size of easily mineralizable organic compartment, hence contribute to estimate the potential nutrients, example, the nitrogen supply by the soil. The aim of this study was to assess the changes in soil health, quality, and functionality following the conversion of native grasslands to arable agriculture by quantifying the POM and other selected properties in the northern parts of the Kruger National Park. The objective was to quantify the POM and other selected soil properties after the native grasslands conversion to banana plantation. To also develop some quantitative relationships between the POM and other selected soil properties under the native grassland to cultivated soils. It is hypothesized that no changes will occur on the POM and other selected soil properties after the conversion of the native grasslands to banana plantation. There is no relationship between the POM and other selected soil properties under the native grassland and the banana fields. So materials and methods, this is the area where the study was conducted with the coordinates and also the elevation of 392 meters above the sea level with the annual precipitation of 600 millimeters with the annual temperatures from 26 degrees to 30 degrees Celsius. And the area is 348 hectares long. The sampling strategy was that the native grasslands, we had 3 pits, and the banana fields also had 3 pits, whereby they have the soil type of false refill. The sampling depth increments was from 0 to 20, 20 to 40, and 40 to 60 centimeters with 3 replications, and a total of 54 samples were collected. So this is how the classification was conducted and the sampling method. So the procedures for lab analysis: the particulate organic matter was done through the wet sieving method. The soil organic carbon was analyzed using the Walke and black ignition method. The soil pH using the electrometric method, whereas the particle size distribution was analyzed using the hydrometer method. A two-way analysis of variance ANOVA was executed to compare the land use types and the soil depth on each of the soil quality indicators measured for each soil using the GenStat 18. Differences between the mean significant factor was assessed with Duncan HSD. The LSD lists significant differences where P is equals to 0.05 was computed to separate the means for all the properties. And also the linear regression analysis was used to determine the correlation between POM and SOC. So these are the results of the pH when it was analyzed with water and KCl, and also the results for particle size distribution under 2 different land uses, which is the grassland and banana. So the graph here shows that It has been a comparison when we're looking at the particulate organic matter under two different land use types, which is the grassland and the banana. So we're looking at different depth from 0 to 20, 20 to 40, and 40 to 60. And it indicates that under the banana, there have been more particulate matter as compared to the grassland, native land. And when it looks to soil organic carbon, here's the figure which compares the soil organic carbon, and it has shows the relationship between the particulate organic matter and the soil organic carbon on different land use types and the depth intervals. So it has been indicated that the relationship, it shows the negative correlation between the two variables. So the significant differences were observed between the pH mean values, land use types, and soil depth. And the pH values under both grassland and the banana were acidic. The texture for both land uses was sandy, with 4 to 12% of clay, 3 to 9% of silt, and 82 to 92% of sand. In comparison to grassland, which has a mean average of 283 average, and the banana has shown that it has the highest average value of POM, because there was a lot of mulching covering the soil under the banana farming. So the POM in the soil is affected by the management that impacts soil organic matter accumulation. And the POM is higher in the depth of 0 to 20 centimeters under the grassland land use. And we're also looking at the subsurface for 40 to 60 centimeters, both land uses, the particulate organic matter begins to decrease. And the soil organic carbon under the grassland also increases as the soil depth increases, indicating that there is more carbon in the subsoil than in the topsoil. And the total organic carbon in soil can be reduced by farming, but does not imply that loss of organic carbon storage in the stable pool. And the negative relationship between POM and SOC confirms the interdependence between these two variables. In conclusion, overall, the impact of land use and native soil POM content on the soil organic carbon was found to be significant, and cultivation produces organic carbon depletion. However, this does not imply sustained organic carbon depletion. And the result shows high proportion of stable organic carbon in soils under the grassland with lower POM content. Even it is highly cultivated land, the organic carbon storage potential under the banana crop can be boosted with good management, such as frequent residue additions, limited tillage, and balanced fertilization. I would like to acknowledge the School of Agriculture, my supervisor, Mr. Mbanjwa VE, and the Baobab Fruit Farm in Guyana to allow me to conduct this study. I thank you.
Thank you so much, Mr. Amma. Can I take 3 questions? The first one.
Hi, it's a piece. Are you good?
Hi.
Are you
I'm
good?
fine. I'm fine, thanks. How are you?
I'm also good. Soil organic matter, it's a complex soil property. It has different fractions. And then I want to know, why did you specifically choose your POM? And then another question is that you said that there is high POM in your banana plantation, but you did not give us the reason as to why. So I want to know, what do you think could have been the reason as to why there was a high POM in banana plantation?
Okay, thank you very much for your questions. The first one, as I've mentioned, that POM is one of the fractions of organic matter. So in this case, POM is one of the easily mineralized or decomposable fractions. So that's why we were able to choose POM as one of the fraction inside the organic matter. And it drives most of the functions when it comes to organic matter in the soil, such as improving your structure, such as also improving the water infiltration, as I've mentioned. So POM is important in that case. And to answer the second question, reason why the particulate organic matter was higher under the banana field is because the banana field, they practice, there are a lot of management practices that are done under that particular land use. So such as the fertilization, such as manure, and also those cover crops that are on top of the soil under the banana plantation, they also increase the particulate organic matter in that particular field. Thank you.
Thank you very much for your good presentation. But I have got, I think, 2 questions. You formulated a hypothesis, but you didn't tell us whether you have accepted or rejected those hypotheses and what the implication of accepting or rejecting. And the second question is, I think you made a very nice recommendation, but how do you make sure that that recommendation is implemented? And who is going to implement it? And do you think that this recommendation will reach the department or the policymakers? Because most of the time, you know, we made recommendations and so on, and those recommendations will go to the backyard grave. You know, the backyard grave is a library. You get your master's degree, and the copies of your master's will go to the library, and no one will see it. Thank you.
Thank you for your questions. The first one, to answer the first one with regards to the hypothesis, yes, I do accept the hypothesis because we were looking at the changes in terms of the conversion of 2 different land uses, and when we're looking at the particular physiochemical characteristics. So it has been shown that whenever there's a conversion from a native grassland to banana field, there's been a change in terms of the soil organic carbon and as well as the particulate organic matter. And again, these 2 properties, they are also interdependence, like they have a relationship. So the other one, which, if the other one increases, the other one starts to decrease. When we're looking at, for instance, when we're looking at the banana plantation, with the POM specifically. So the POM when it's converted to banana field, it started to increase because of the reasons that I've already mentioned earlier on. And the soil organic carbon started to decrease. So it means that we accept this hypothesis. And then to answer the last one, whereby you asked about the recommendations, is that the recommendation stated that all these properties or the findings in this study, they can be improved through management practices. So mainly the farmer or the farm owner who is responsible for that farm is the one who's going to, we're going to be able to explain these recommendations and also to tell him why. And again, we are able to identify negative environmental impacts that can occur if he doesn't follow these recommendations. And I think that they can also be adapted to all farmers, not only in this particular farm. Thank you.
Thank you so much, Mr. Tluwamma. Can we give him a round of applause? I'm going to hand over to Dr. Mdleje to continue chairing.
Thanks to my co-chair, she has actually made my life a bit easier because now I'm more calmer because she started the session. Thank you so much. Yeah, before we can go any further, I'm Zolethu Mdleje, so I'll be chairing from now onwards. Yeah. Before I call the next presenter, I would like to check if Zinhle Nkosi is around. Is she around? Okay. In her absence, then I would love to call Patience Ramabale to come on stage. Patience Ramabale. Thank you.
Um, Good morning, everyone. My name is Patience Ramabale, and I am supervised by Professor E.M. Zwane and Professor A. Belete. I will be presenting to you today a portion of my PhD study, and the portion I'll be giving to you, it's just the determinants of social media use for smallholder farmers in Limpopo Province, South Africa. Just to give you a little background of what I am doing, my study is mainly on social media and its use around the province. So I had several objectives, and just to mention a few to you, I had the opportunity to describe the effectiveness of social media around the province by smallholder farmers, to identify their needs, to determine the social media platforms that they are using, and also to explore the perceptions and develop a model for social media use in the province. I just hope I'm going to have enough time to present everything to you. So the introduction of my study as projected there, we all know that agriculture is a very important sector in South Africa. But are the farmers in this sector satisfied? The farmers face several challenges, and some of the challenges that are facing includes extension services, low productivity, and also little information access. And that is where now the core concern of my study is. Are we going to have productive farmers if they are less informed? So we've got social media that is emerging at a very fast rate now, and I see it as an opportunity for farmers in our province to harness from it, to get something very good from it. So while the adoption is practiced widely in other countries, there is little that is done in our province for our farmers to adopt the use of social media. By so saying, I'm not saying there's nothing completely done. There is just little documentation and little formalization of the use of social media. We are all aware social media is being used. Yes, farmers are using social media. Extension officers are using social media. Is there any guiding policy for them to use social media? Are there any formal guidelines in place? You know, are all acts in place? So those are the lines that I'll be going through. But as I mentioned earlier, my focus for the sake of this presentation and the 10 minutes will only be on the determinants, which are the socioeconomic characteristics. To achieve the results of my study, I was it was a quantitative study. In the Limpopo province, I included all the districts for the sake of fairness and inclusivity, and to avoid a naive generalization. It included 420 smallholder farmers, and I used a semi-structured questionnaire to get the responses from the farmers, and it was cross-sectional. This is, I just conducted the study at once, so I did not have different phases to get the expected outcomes. Then the data that was analyzed, the data that was collected, was analyzed descriptively, and the software that I used was SPSS. So as you can check there, I won't be going through the details one by one, but I just highlighted that which should be of utmost importance for you, for your knowledge. If we check the gender of these farmers with regards to social media usage, of course, we've got the highest, which were females, which is good. 237 farmers were females in my study, and around only 183 were males. But if we check in the district, in Mopani district, scored the highest number of males, and the highest number of females were spotted in Sekhukhune district. So in relation to gender, it is very important to consider gender dynamics, especially with the use of social media. You are aware that this particular usage or action will require knowledge for those that are less knowledgeable or illiterate. So gender dynamics are very important, and as well for the development of the program, such that whoever is going to be responsible to develop the programs and whoever will be responsible to organize the meetings and everything should take the issues of gender into consideration and check that everyone is included, including We are counting both males and females in the province. Then with regards to marital status, a bit sensitive now because I've already went to the people and, collected the data. In our culture, especially in Limpopo province, marriage is one of the most sensitive issues. Just to go through the numbers before I take you through the details. About 40 farmers in Mopani district were found to be married, which is the highest number of married people in all the districts of Limpopo. Then Capricorn scored the highest with the divorced ones at 33, and the Capricorn district again scored the highest with the number of widows, and Mopani district scored the highest with the number of singles. So now, as I said, marriage is a very sensitive issue, especially in our province. With regards to the issues of information dissemination, developmental programs and other things, take it for instance, married people or married couples have got more opportunities than the single and the widowed, or the widowed and the divorced. The reason here is when you are married, you are better off with someone. You make combined decisions, and you are better advised. If you're about to do something, there's someone to advise you. So now, literature shows that the divorced and the widowed are marginalized, especially in our rural communities. For example, if you find a widowed woman in the community, farming communities especially, they are, I cannot say they're discriminated, they're isolated. There are activities they cannot do, and there are activities they can do, especially where they have to mix with other people. So when you check, other studies are also confirming that the widowed and the divorced women face economic and social vulnerabilities. These are just some of the few that I've mentioned. So in terms of age, across all the districts, Bembe district scored the highest mean in terms of age. This says in Bembe district, most of the farmers were old. Then came to Mopani district, then the mean age there was the lowest at around 34. Then it's just showing that in Mopani district, most of the farmers at least were a bit younger. With regards to social media, age is also a very important issue, because in literature it's indicated that younger farmers are likely to use social media more than the older farmers. But in my study, as you can see, most of the farmers are fairly old. But will they be left out in terms of social media use? No, they cannot be left out. So that's why we're talking of age and how it's affected by the social media use. So we've got three researchers there that are saying, the first one, Mbungwa et al. Reports that older farmers are less likely to use social media, while Vasumati and Arun, 2021, are saying younger farmers are likely to use social media because they are able to use technology and they're interacting with many peers. It becomes very easy for them to start using these particular tools. Sorry. Then in terms of qualification there, around all the districts, the significant ones, I just highlighted them in green for you not to go through the whole table. So most of the farmers in this study has had a secondary education, whereas just at around 41, which was the lowest, they had ABET, which is the informal qualification that you acquire if you were unable to complete your matric. So in terms of education, apart from just skills that they develop or the literacy levels that improve, in terms of education, it is believed that the more you further your studies, the more people you get to interact with. If you check your cycle while you were still in high school or primary, and your cycle now that you're in university, the likeliness of your cycle that it has increased is very high. So you are likely to learn many things when you are surrounded by many people, unlike someone who has not gone far and only knows the people they went with to crèche or primary day. So We talk of the monthly income, which is just the general income of the farmers. Most of the farmers had their income less than 3,000, while only a few had their income above 6,000 rands. What does this tell us about the farmers of the Limpopo province is that when it comes to economic opportunities or economic development, they are still left behind. And as you might know, that social media would require, for you to use social media, you would require some money to buy the tools, right? So if their income levels are still low, some of them, even though they would wish and have the knowledge, it will be very difficult for them to buy cell phones and also to afford data. Then these are just the results on the number of family members. The number of family members in my study was very important as well, because I believe that if we are a family of 5 and I've got a social media device tool, a cell phone, and he does not have a cell phone, he can use my phone to access the information that is out there, or use the cell phone of so-and-so. So the more the number of people are in a particular family, the higher chances of social media usage in that particular family. Then just in conclusion, that this study aimed to understand the use of social media. The focus was on the socioeconomic characteristics, and it revealed the importance of considering socioeconomic characteristics in designing interventions for small-scale farmers. Age, gender, marital status, educational level, and income can significantly influence farmers' access to resources. Then targeted and context-specific approaches are needed in this regard to address the challenges of small-scale farmers. These are the recommendations from my study technology training programs by the Department of Agriculture and Extension officers for the farmers, such that they are well equipped with the basic knowledge of using social media platforms, and also a social media communication model that my study has already developed in this case. Just to take from what Prof. Belete asked the previous presenter, that who is going to see this model and implement it? I have already secured an appointment with the Department of Agriculture that upon the completion and confirmation of my study, this model is going to be presented to the Department of Agriculture for approval, and of course I'll be available to discuss this model and guide in implementation. Unfortunately, because of time, I can't be going through the model, but this is the model. I am available anytime for the discussion of this model. These are the references that I used. And before I thank all of you, a special thanks to my supervisors for guiding me through my research. Thank you.
Thank you so much, Patience. Just a quick one. I will take one, not more than 2 hands. Okay, I can see a hand down there, and then I can see a hand there at the back. Yeah, unfortunately, if then it's more than that, let me just, let's leave it outside the hall for you to discuss with the presenter, because then, we don't have enough time.
Thank you, Patience. You are a very confident presenter. I do, however, have a comment. You presented a lot of the demographics, but you didn't link it to social media use. So we cannot see, because there are various studies to say that older people are actually more inclined to sharing disinformation, misinformation, the over 60 group. They are actually also very active on some of your social media platforms. You simply mentioned your variables and you made assumptions, but you didn't show us how it linked in your study to actual social media use.
Okay, let's is this a comment or a question, ma'am? Oh, it's a comment. Okay, thank you so much. I hope you picked up the comment. Can I take that last one? But then, yeah, our adjudicator also has a question. I would allow our adjudicator to ask a question.
Thank you, ma'am, for very interesting topic. And it's also very relevant for our time. But I want to ask you a very basic question. You are talking about social media. And what is social media? If you ask the people here, our definition will be very different. So what is the scope of your definition when you talk about social media? Thank you.
Mine, apologies for taking your time. I'll be very quick. Mine is linked with the first question, but in particular the marital status. I'm just interested, how does the marital status has to do with, or how do you link it with the use of social media? I mean, someone who's a widow, for instance, versus someone who's married, if you can just assist.
Thank you very much for the questions and thank you very much for the comment. It is taken into consideration. I will start with the first question asking what is social media, and in this case, what is the scope of social media? Social media is a mass communication platform that is used for dissemination of information. I know most of us are using social media for different reasons. It could be for entertainment, it could be for job seeking, it could be for networking. But for the sake of my study, social media, it's defined as a platform for disseminating agricultural information. And for the scope of this study, I'm using social media as a platform where farmers and extension officers can meet and share ideas. Hence, I developed that model that would show how farmers would send messages to extension officers and how extension officers will send the messages. These are agricultural related messages to the farmers, farmer to farmer, extension officer to extension officer, and all the way around, making a cyclic model. Hope that had answered your question. Then the second question, how does marital status link with social media use? In my study, when I'm talking about marital status linkage to social media use, I will give you a small scenario. There are two farmers. One farmer is able to use social media, and one farmer who is a widow is unable to use social media. The other one who is a widow and cannot use social media, the Department of Agriculture can organize programs for training on social media use. Then you find that the one who is a widow is unable to attend the programs due to the cultural restrictions that exist in the farming communities. So hence I am talking of marital status, that sometimes the ones that are married are having more advantage than the ones that are not married. And in terms of affordability as well, we would assume that sometimes the widowed, because of not having partners and not having combined income with who was supposed to be their partner, they are unable to afford and, just unable to access more information. Most of the information we have, mostly if you've got a partner, sometimes most of the information you get it from your partner, and in the absence of the partner, there is no information.
Unfortunately, I have to then, to allow the next presenter because now we don't have time. Unless, doctor, you still have a follow-up, it's okay. Yeah, with regards to you, Ms. Porter, you can actually continue outside in maybe advising on some of the things, maybe some of the models in the statistics that can link social demographics and social media so that it can actually improve her work. Thank you so much. Let's give her a round of applause. Yeah. Right now, I would like to call Sako, Tabang Sako, to come on stage and present.
Good morning, everyone. I'm Travan Sako, under the supervision of Prof. T.L. Jasi and co-supervision of Prof. J.W. Ngambi, presenting a study titled Impact of Coat Color on Body Height and Length of Bapedi sheep. This is the presentation outline of the study. Bapedi sheep is a tough, disease-resistant and fertile breed South African indigenous sheep. It originates from the semi-arid regions of the Limpopo province. It is titled Bapedi sheep because it was first reared by the Bapedi people. Body measurements have been used in ruminants to estimate body weight as well as breed description and characterization. Coat color, which is a genetic factor, is known to adapt animals to different climatic zones and has considerable influence on the performance of various stocks. In the natural environment, dark or brown coated colors are preferable, whereas for economic purposes, pale, i.e. white and yellow, are desirable. So looking at the pictorial presentation is a demonstration of what a Bapedi sheep looks like. It's a typical felt and long-tailed and also multicolored indigenous type of sheep raised in South Africa. So a coat color is an important economic trait in sheep breeding, and dominant white color of sheep could be best adapted to the local environment and preferred by the farmers. So smallholder farmers in rural areas, on the other hand, find it difficult to estimate live body weight on their animals due to lack of weighing scales, which may be expensive to purchase due to financial constraints. Animal coat color as one of the major breed characteristics is an important production and economy trait, and studies propose that sheep coat color may have correlation with reproductivity, morphological traits, and energy homeostasis. So given the economic importance of the trait as well as its importance for sheep breeders, there is a need to broaden our knowledge of the genes which are involved in establishing a particular coat color pattern. So there are several studies that investigated coat color in sheep. However, there is a need for more information on the influence of coat color gene on morphometric traits of extensively raised sheep. So the aim of the study was basically to evaluate the impact of coat color on body height and body length of the Bapedi sheep. So the objective simply by determining the impact that the coat color has on the body height and length of the Bapedi sheep. The hypothesis is in line with the objective. So with regards to the study size, the animals are populated throughout the province, but then mostly your indigenous Bapedi sheep are under the Bapedi sheep breeding society, whereby most of them are found in the Sekhukhune district. So in terms of the number of the animals that were used in the study, a total of 636 Bapedi sheep were used. However, we used what we call a multi-stage sampling, whereby we started with the breed itself, then after went on to the breeding society, then on went to the farmers. While we are at the farmers, that's then we purposely selected them and then used the census approach, whereby we used all the animals in the kraal or the population. So all the sheep that were used in the study were managed extensively so. And then cross-sectional design was used in the study, which basically means data was collected at once. In terms of the data collection, the body measurements were taken from the animals in a standing position with the head raised as described by Food and Agricultural Organization. Morphometrical traits were measured using a Taylor measuring tape, which is in centimeters, and then the qualitative traits were observed based on the guidelines from Food and Agriculture Organization document. So in terms of the data was analyzed using version 26 of the SPPS software. So Spearman correlation was used to examine the association between the qualitative and the quantitative traits. So all the statistical analysis were performed at the 5% significance level for statistically significant and 1% for highly statistically significant. So the following general linear model was used for the study to determine the effect of the coat color trait on the traits. So the results are as follows. Table 1 shows the variation results of effect of coat color traits on body height and body length. So we find that there was statistical significance when it comes to body height in females and body length in females when coming to the coat color pattern. However, with regards to the head color, body color, there were no significance in terms of the females and also with regards to the males. So we went on to do the means for group in terms of the homogeneous subsets for the body height in the females using the Duncan method, and we found that there were no significance within the different groups. And then also for the mean subsets for the body length. So first one was the body height, then the second one was for the body length, following the statistical significance. So coat color pattern had a significant effect on body height and body length in females, whereby the multicolored coat pattern had the highest effect, followed by uniform multicolored. So this means that farmers can select multicolored to produce animals with higher body height and longer body length, which basically means the bigger animals. Color of the body and color of the head had no effect on body height and the body. So the current study disagrees with the study that was conducted in 2017, which reported that interaction effect of coat type and coat color exerted no significance on the zoometric traits of West African dwarf sheep. The current study concludes that a positive relationship was found between coat color pattern and body height, as well as body length of Bapedi sheep. Therefore, body height and body length of Bapedi sheep might be improved by selecting for coat color patterns. The findings suggest that coat color pattern can be used to predict body height and body length of the Bapedi sheep. It is recommended that more studies be done on the influence of coat color and coat structure on morphometric traits of sheep for improved selection by the farmers. So further investigations are needed on other breeds and species to identify the qualitative traits that can be used to predict economically important morphometrical traits. So I would like to acknowledge the University of Limpopo Department of Agriculture, Economics and Animal Production for offering me the opportunity to conduct the study, Limpopo Department of Agriculture and Rural Development for support and data collection, Prof. T.L. Chasi and Prof. J.W. Ngambi for their support and for their supervision, then the Chasi research team for their support. So these are some of the references that were used in conducting this study. So thank you.
Thank you so much, Sago. I think we have enough time, more than enough time for questions. Yeah, any questions, comments? Okay, I'm sorry. I think you'll come after.
Okay. Thank you, the presenter. In your presentation, you highlighted the issue of coat color, but in your discussion now in the results, you are showing the coat color patterns. What's the distinction between the two, the coat color and the coat color pattern? That's the first one, as it will be influencing the linear measurements trace that you'll be measuring. Right. And going to the second one, you showed us the results that indicated that the coat color pattern influences these linear measurements in females significantly than in males. What could be the basis for this?
Okay, thank you, Mr. Sago. This is a good and interesting study, so I just want to be clear on something. If you're having a black goat and a white goat, and then the results that you find there, to say the white one has this body, more body height, how sure are you that the result that you get is not from the type of feed they eat. Thank you.
Okay, I'm not a breeder, but my question it's exactly the same. I was going to ask somehow different to say, how did you account for nutrition on body height and length, which may vary from one farmer to another? Particularly, you stated that they were, extensively kept, but they may be supplements, maybe summer leagues or winter leagues, or whichever that may. So how did you account for that?
Thank you for the questions. I'll just speak here. With regards to the first one that speaks of the coat color and the coat color pattern, the distinction between the two, the coat color is basically what's this? A main heading for a group of traits. So in terms of coat color, under coat color we have the coat color pattern, we have the color of the head, we have the color of the whole body. We can also include the color of the legs and all those things. So that is why then now in my results I spoke of coat color, but then the results I spoke of coat color pattern, color of the head, color of the body. And then with regards to the second one, the males versus females one, Bapedi sheep is an endangered breed. So with that being said, mostly is because of the fact that a lot of people that have it usually use what's this breed again? The Dopa. Use Dopa rams on the Bapedi sheep. Use just mainly for the fact that they are able to adapt to the conditions or to the regions of this province. So they need those adaptive characteristics in their offspring. So that is one of the leading causes of the depletion of the breed. So in terms of the males versus females, most of the Bapedi sheep breeders, once they get a male, they don't want to keep it, so they sell it. That is why now then the numbers are low in terms of the males kept versus the females kept. So that is why now then we find these numbers being like this. And then in terms of the black and white goatee, and then how am I sure of the feeding regime and all those things? Honestly speaking, we did not account to the feeding regimes because now we're speaking of different locations and all the districts within the province. So that can be added to the next study. Thank you.
Thank you, Sako. Unless, Doctor, you have a follow-up question. Do you have any? Okay. Do you still have enough time? Do you still have time? Okay, you still have a follow-up. All right, it's okay.
Just as a follow-up, to what extent then, by not including or factoring nutrition, to what extent are you confident with your results, given that nutrition is also one of important factors? How confident are you with your results?
I can't necessarily, how do I answer this one?
Okay, just a quick one. Maybe you can mention that as a limitation of the study because nutrition is very important. Just a quick one.
Mine is just maybe a comment from the previous question which they've just asked, where about your heading, your title actually. Don't you think if you were supposed to say color patterns. Because if you go to your results, you have actually talked much about the color. If I'm not wrong, I'm just going to your title. Your title, Mr. Swafu, it seems it was more of a, you say, code color. Can you maybe put a pattern so that you can cover, your title must cover your results? It's a comment, I'm not saying change it.
All right, thank you so much, Dr. Silepe, and thank you so much, Saku. If there are any follow-ups, it's fine, you can actually communicate with him outside the hall. Otherwise, let's give him a round of applause. Thank you. Our next presenter now, it's Swafu. Swafu, are you here? Okay. Please come forward. It's fine. Yeah, it's fine. It's fine. Yeah, I think it's fine. It's not a problem. Until then he comes back. Then we'll be fine. Before Swafu presents, I just want to check if Zintle Nkosi has she arrived? So that when we conclude, before we conclude, we can give her a platform to present her work. Is she here? Okay. All right, it's fine. Now you can start.
Greetings to you all. My name is Michael Swafu. I'm here to present a study which I've done with my co-author, Peshia Dlamini. This is part of a big project which I did for my master's project. And then the title of the study is Using Classical and Geostatistical Insight to Unravel the Spatial Variability of Soil Nutrients, a key step in establishing site-specific nutrient management. In our country, smallholder farmers contribute significantly to food production, and they play an essential role in the nation's food and nutritional security. But soil quality in smallholder farmers is progressively declining. And the reason behind that is that farmers, smallholder farmers, they don't know their conditions of their farms. What do I mean by that is that farmers, they don't know the different soils that are underlying their farms, and they also don't know how variable are the soil nutrients which are in their respective farms. These are the factors which are affecting banana production in smallholder farmers. And they are categorized into land and also soil characteristics, which is poor soil fertility, soil variability, and also soil degradation. But there are also other factors which are related to the environment, which is temperature and also rainfall. To address the problems, we did a study where we were unlocking the land capability, soil suitability for sustainable banana production. In this study, we're trying to check if the land characteristics, are they capable for agricultural production? What we found is that in as much as some portions of the farm, they are arable, but then they are marginally arable, which in a way affected their productivity. And then we went further, and then we tried to check how the soils are suitable for banana production. Here, we were linking the soil properties with the ones which are required by the banana crop. And then what we found is that they are suitable, while others they are not suitable for banana production, which basically means that these farmers, they were indeed utilizing their land without considering land evaluation. But then we were left with some answered question, and then we went further into now checking the soil nutrients which are essential for banana. We wanted to check how are they variable across the banana plantation, and then we also wanted to delineate the management zones, and then this led to the study in question. The objective of this study was to assess the spatial variability of the essential soil nutrients, and also to delineate soil management zones in the banana plantation. The study was conducted at Makuleke Farm, a smallholder farm in Collins Chavan, a local municipality, which is in the Vembe district and in the very same province that we are at. And then to assess the spatial variability, we adopted a grid soil sampling strategy where it helped us to collect representative soil samples for fertility assessment. And then we managed to collect 27 soil samples across the 12-hectare banana plantation. And then these are the physical-chemical properties which we analyzed from the collected representative 27 samples. And then to assess the spatial variability of the soil nutrients, we adopted geostatistics analysis, wherein we used regression kriging to assess the spatial variability, or to draw the spatial maps of the nutrients. And then we used regression kriging to interpolate between representative points across the farm. And the reason behind that, the reason behind us using regression kriging was that we also wanted other covariates which could help us to predict the soil nutrients across the farm. And then we also used the spatial maps to delineate the soil management zones across the farm. And then, as you can see, this is a map. These are maps of the essential soil nutrients, which are telling us that indeed the nutrients were spatially highly variable across the banana plantation. And then we went further into now delineating soil management zones, which were to help the farmers to categorize their management practices according to the variations of the soil nutrients which we found in the farm. And then it is evident that there is a huge variation of soil nutrients across the plantation. But then the farmers were continually homogeneously managing the field, and that led to under application of fertilizers, and in other areas it led to over fertilization of nutrients. And then what we learned from this study is that before any agricultural practices could take place, there's a huge need to assess land evaluation, and then there's also a huge need to assess the spatial variability of the soil nutrients, which they are essential for management practices, to allocate management practices to specific locations of the field. And then I would like to acknowledge my co-author, Professor P.E. Dlamini, and I would also like to acknowledge Mr. V. Mbanjwa and Ms. R. Chauke, who helped us to classify the soils across the farm. I would like to acknowledge the University of Limpopo, and I would also like to acknowledge the National Research Foundation and also Makuleke Farm for allowing us to conduct this study.
Thank you. Just on time. On time. Let me open a platform for questions and answers. Yes.
Thank you for that wonderful presentation. My two questions are not that difficult. According to your study, what do you define a smallholder as? Because the term smallholder is actually an umbrella term, according to some of the studies I have researched about. And then the second one is that due to the term smallholder being an umbrella term, do you think it has some sort of effect towards your study in terms of the farmers' perceptions?
Please repeat the question. You have two questions or it's one?
It's two.
And then please repeat them, because...
Okay.
Yeah.
The first one is, what do you define a smallholder farmer as in your study? The second one is that, do you, in your own opinion, think that the term smallholder farmer, does it not have any influence on the farmer's perception of the contents you were just providing us with?
Oh, okay. In my case, the smallholder farmers, they are resource limited farmers in the rural areas. Normally, they are planting crops largely for their consumption. And then in terms of the perception, even though I don't know how it's related to the soil science discipline where I'm in, I don't know how to link it, but I don't think it has any effect on what I was trying to do here.
Thank you. We'll just discuss it outside as well.
Okay.
Okay. There's a hand at the back.
Thank you, Mr. Swafo. I've just gone through this study of yours, and I'm aware that you have already published. So, just an interesting question from me is not to do with the study, because it seems you have done it. It's published. My interesting question is, did you ever went back to the farmer for when you have done this special testing, whether there is nutrients enough, not enough? Have you managed to go back to him and then give an advice how must he do the fertilization and irrigation part? Thanks.
Okay. Yes, I did go back to share with the farmers the results that we got. And then I also tried to show them how they can easily manage their farm to get optimal yield. But then in terms of irrigation, it wasn't really my focus. My focus was on the soil properties.
Thank you, Mr. Swafo. First, let me commend you on doing a very good study as well as getting published. My question, maybe it can be a follow-up from defining what a smallholder means. In your view, you have recommended that they do site-specific management. Are they in a position to adopt that, in your view? Can they do it?
In my view, them alone, I don't think they can do it because some, they are illiterate, so they would need intervention from us, the scientists who know how to do the work, and then they will also need intervention from our Department of Agriculture to then now help them to get the relevant people to guide them on how best they can manage their farms.
Thanks, Mr. Swafo. I'm just wondering whether if you have data on production and maybe quality in relation to, you know, the soil nutrients that you have indicated that, you know, it varies across, and the application of fertilizers make things worse, but even if it's for future. But on a more practical part, which relates to a question that has been asked, how feasible or how practical will it be for these farmers that you are working with when now they are applying fertilizers to apply the correct amount in the right space? Because there's quite a lot of variability, as you have showed. Now in practical terms, given your recommendations or your results.
Okay. The reason there was behind this studies was initially we went there and then we noticed the spatial. The second question, which is saying, is the practicability. What you can notice from this study is that this 12-hectare plantation is underlaid by 4 soil types, which were mostly controlling the nutrients. And then it's going to be much easier if then now the management practices, they are allocated based on the portions that are covered by the different soils.
I hope you are content, Doctor, from the response. Anyway, you still have a follow-up? Okay, all right. Anyway, because of time, let me just say thank you to you, and can you please give him a round of applause? Yeah, now we are going to our last presenter, which is Tendo Mugwena. Can you please come forward? Tendo, oh, she's coming.
Morning, everyone. My name is Tendo Mugwena from the Department of Geography and Environmental Studies. I will be presenting a study that is ongoing on comparison of different non-parametric classifiers and the majority voting rule to differentiate tamarix from surrounding vegetation. This is under the supervision of Professors Dau, Newete, and Abu Taleb, and the mentorship of Dr. de Klerk. Tamarix is a plant commonly known as salt cedar. It is a shrub or a tree that can grow up to about 12 meters. It flowers and has pink or white flowers depending on the type. The leaves are scale-like, and it has very specialized salt glands that are able to take out salt from the soil. And so what tamarix was introduced, and this is just a map showing the distribution of the density around the world. In South Africa, the distribution is shown in the map as follows. We do not currently know when exactly it was introduced, but we have 3 confirmed species. The Tamarix usnoides, which is native to South Africa. We have 2 exotic ones, the Chinensis and Ramusissima. So as you can see from the flowers, they're quite pretty to look at, and the assumption now is that when the tamarix was brought in, it was because people wanted to have beautiful gardens, but also it was then used in mines for phytoremediation. What we are now finding is that the indigenous tamarix, the usnoides, is hybridizing with the 2 exotic ones. So they're causing a new plant, the one in the middle. And we are able to differentiate the plants based on the flowers that they give out. Because of the specialized salt glands that tamarix has, it can be used to stabilize stream banks, and it also has this extensive root system that extends deeper. It's also been used in phytoremediation and soil suppression after mines have been closed. This shows the exotic tamarix inversion in one of the study sites that I will show you. And as you can see, it has just taken over, the exotic has taken over along that river bank. So, just like any exotic, it causes decline in ecological functions, displacement of native species, and the reduction of water resources, which we definitely need in South Africa. So it brings us to the importance, especially in South Africa, because we have the 3 types, and one of them is native to us. So we do not want to get rid of the native. We want to get rid of the 2 exotics. So when we monitor the extent of tamarix, it helps us determine the changes that it's causing to the ecosystem. We need to remove the exotic and the hybrids because they cause problems. They take up water, they change the ecological system. There have been traditional methods of monitoring any invasive species that involve field inventories. However, because there is a demand at larger coverages, we need methods that are cost effective both in time and money. And this is where we use remote sensing and spatial modeling, which provides us with a monitoring at a broader scale that is cost efficient and also saves some time. Across the globe, there's been various studies that have applied remote sensing for tamarix, and I've just listed some of the key studies done. What we've come to in current research in remote sensing is the use of machine learning. And with the growing utilization of machine learning classifiers, we get various algorithms that are computer-based tools that explain data and statistical analysis. Those algorithms enable the identification of unknown patterns and relationships within dataset values before we include them in models. So for this study, what we aimed to do is to compare various machine learning classifiers to separate tamarix from surrounding vegetation. We wanted to know which is the best machine learning classifier that can distinguish tamarix, and also if we combine the various algorithms or the various models, do we get better results in distinguishing this tamarix? So the study area of the broader project, we chose 4 sites with known tamarix invasion in the Western Cape. The study sites are along riverian areas next to Liuhamka, Dwaika, Prince Albert, and De Rust. What I'm going to present today, however, are results from one study site. The study site that I'm presenting just has the exotic tamarix. So it has the one that we need to get rid of. Of the 4 study sites, only the one along Liuhamka has the native study sites. The Prince Albert and the Dwaika ones have the exotic, a mixture of exotic and the native and also the hybrids. The data that we used was the remotely sensed Sentinel-2 data set that we downloaded from the USGS study sites. We downloaded it for the same time that we did field verification, which was in 2019 November. The Sentinel has 12 bands, but because of their resolution, only 4 bands were extracted. So we only extracted the 10-meter resolution bands, which are band 2, 3, 4, and 8. For the same period, we collected GPS points that had coordinates of the tamarix and other vegetation species that we could find on site. So this is just a flow diagram that shows the analysis. So after the collection of the data, we did a feature extraction in this instance, and for this study, the feature extraction only includes the bands that I mentioned that we took out and used them. But in another objective, the future extraction including vegetation indices. And then the collection of the training site data, the GPS points that I spoke about, we had to include more from the computer. We used aerial photographs to get more points of tamarix and surrounding land cover. And then that data set was split into a 70-30. I'll talk about that just now in the next slide. And then the algorithms that we used the support vector machine, random forest, that is wrong, the nearest neighbor should be the neural networks, excuse me for that. And then we used a decision tree. When we're done with running those models, we combined them into an ensemble model to get classification results, and then we validated our results. So this are the distribution of the training points, which we split into a 70% for the training and 30% for test set. Right, what we got, this is just a visualization of the various model. So, as you can see, the green, the darker green, is the tamarix, and this is now the focus. What we are, from just looking at the maps, already what we can see is that on the top left, where it's highly green, there is some confusion there. We know this because we've been in sites. So, but I'll explain how that came about. So this is now after we've mixed the models. So the evaluation of the metrics. This is how we decide which one is better and which one is not. I can say from just visualizing, I can already see which one did better, but we need the statistics. So we used a confusion matrix, and in our confusion matrix, we used true positives. This is actually where the model identify tamarix as correct. So it correctly predicts the tamarix. And then false positives, the number of instances where the actual clause is negative. So for instance, if tamarix was then identified as vegetation, other vegetation, or as bare land. And then the true negatives, this is the number of instances where the actual clause is negative, and then the model predicted it correctly, so there is no tamarix. Then the false negative, the number of instances where the actual clause is positive, but the model predicted it as negative. Right, I'm not gonna go into what each of those metrics is for the sake of time, but we then calculated the accuracy of each model, the precision, the recall, as well as the F1 score of these various models. And just to quickly go through the performance. In this graph, I used the overall accuracy, which is out of 100, and the line shows the kappa coefficient out of 1. When we did the overall accuracy, we found that the ensemble, the majority voting, did better than all the other models. However, when we then really evaluated the performance based on the other matrices, the precision, the recall, and the F1 score, the majority voting is not doing as well as the support vector. The precision for the support vector was higher than the majority vector, majority voting, as well as as well as the random forest. I will skip to, this is just the summary of the exact numbers. So I'm going to skip to the conclusion as to why the overall or the majority voting did not do well with the precision, recall, and F1 score, and what would be the effect. So we choose the classifiers based on what we want to do. And if we have a high precision, then it minimizes the false positives, where the models like support vector machine and random forest did very well. And then if a balanced performance is desired, the decision tree and the neural network should be considered. But in our case, they didn't do as well as the support vector machine and the random forest. The ensemble methods, the majority voting, provides a balanced trade-off between the precision, recall, and then the overall accuracy, because it leverages the strength of the multiple classifiers, even though it does not do well, or it did not do well in precision individually. So going forward, the choice of the models will be specific. So when we go to the other side on what we want, because with the other side will be taking up, differentiating the native from the exotics. Thank you.
Thank you so much. Just a quick one. I'll allow one because then we are in the time for whereby we have to break. We'll just take a quick one. Okay.
Okay, thank you very much. Thanks for the presentation, very informative. Mine is just a comment on your use of the word exotic. Yeah, the word is correct. However, in terms of exotic and invasive. So invasive is a type of exotic. However, when I looked at your result, at your, the problem that you found there was that this is more of a nuisance in the country. So once it becomes that, it's preferably to call it an invasive rather than an exotic.
Noted. Thank you.
Okay. I think I would just, okay, just a quick one, quick one, because then now we are in the time whereby we have to be breaking. Just a quick one.
Okay, thank you, Chair. Thank you for a very calm and collected presentation. I will put in two questions. You can answer it the way you like. The first question is, what is this limitation in terms of area? You can relate it to the resolution. Second one is, I heard you talk about the confusion matrix, but in terms of data, you only said 70% for training and 30% for testing. Where did you get your validation from? Did you do cross-validation or you had another data? Thank you.
Okay, thank you. The first question, let me answer the second one while I still remember it quickly. We collected the field data for validation, but because the classifications were run in R, we combined the field data and the other training points that we collected manually using aerial photographs, so that the model or each algorithm splits it randomly. It has been shown that it avoids the user bias if I split it myself, whereas if I just combine all the training points and say split into testing and training. The first question, quick reminder, please.
Okay, what's the area limitation? What's the smallest area you can identify using this imagery?
It's a 10-meter resolution, well, the bands that we used. So the smallest will be 10 meters on the 4 bands that I used for the study. But it goes up to 30 bands, and it can also be improved if we pen sharpen the bands.
All right. Unfortunately, I will have to then cut it short because of time. Then if you are discontent, you can actually advance the discussion outside just now when we have a tea break. Ladies and gentlemen, this marks the end of this session from the chairing team. Thank you so much. I was asked to pass this quick one. Our tea break is going to be 20 minutes, and those who will be presenting, they were asked to come and upload their presentations now. Thank you.
please approach the podium. The general rules are still applying. We have 15 minutes allocated to a presenter, comprised of 10 minutes of the presentation and 5 minutes of responding to questions. Right. Okay. Thank you.
Good morning. My name is Victoria Hlokwe. I'm a PhD student in animal production under the supervision of Prof. Jassi and Prof. Mbazima. I'll be making a presentation under the topic Expression of Growth Differentiation Factor 9 mRNA in pre-ovulatory ovarian follicles of Potchefstroom Kokok chicken breed. The slide there is just displaying the outline of my presentation. Potchefstroom Kokok is an indigenous chicken breed that is used by the farmers in rural areas for the production of both meat and eggs. This chicken breed can survive well in free-range conditions with less intensive care, and this is due to its good adaptation. There's a process called gene expression, which is just the way in which information that is in a gene or DNA is used to produce a functional gene product. Eggs are the most important forms of animal proteins in the world, and due to an increasing human population, the demand for eggs is high. However, Potchefstroom Kokok chicken breed has low egg production, which leads to reduced egg yield, which in turn affects the profit margins of the farmers who are using it. Therefore, improving the rate at which this chicken is laying might play a vital role in improving the egg yield and in turn the revenue of the farmers. Studies published previously have shown that improving the chicken ovarian follicles have enhanced the egg production traits of the breeds mentioned. And the paper published in 2005 have shown that GDF9 mRNA was found to be expressed high in the small yellow follicles of the breed mentioned. However, the GDF9 mRNA expression in the ovarian follicles of the Potchefstroom Kokok chicken breed is unknown. The study aimed to improve the egg production of Potchefstroom Kokok chicken breed, and the objective of the study was to determine the expression levels of GDF9 mRNA in pre-ovulatory ovarian follicles of Potchefstroom Kokok chicken breed. This is just the hypothesis that is in line with the objective. The study was conducted at the University of Limpopo Experimental Farm and at Animal Production Laboratory, and it was also conducted at the Department of Biochemistry, Microbiology and Biotechnology Laboratory and at Ingaba Biotechnology Laboratory in Pretoria, South Africa. A total of 3 chickens were randomly selected as recommended by the paper that was published in 2005 and 2021. The hens were raised under intensive production system according to the normal husbandry procedures as described by the paper published in 2012. A completely randomized design was used. The chickens were slaughtered following the procedures of the paper published in 2017 for harvesting the ovarian follicles with sizes F4 to F1 for determining the GDF9. We have the picture of the lab where the mRNA was extracted. RNA was extracted from frozen ovarian follicle tissues using the RNeasy mini kit according to the manufacturer's instructions. And the extracted RNA samples, they were sent to Ingaba Biotechnology Laboratory for gene expression analysis. Statistical package for social sciences version 28 was used to analyze the data, where students' t-test was used when the target gene and control gene were compared after confirming the distributions of mRNA expression. ANOVA was used to examine the differences in the relative expression levels of GDF9 in pre-ovulatory ovarian follicles of the breed. Duncan multiple range test was used for mean separation, and this is the model that was used to achieve the objective of the study. These are the results of the study, and figure 1 is showing the levels of GDF9 mRNA in pre-ovulatory ovarian follicles of Potchefstroom Kokok chicken breed. The findings of the study showed that GDF9 gene mRNA was expressed in all the studied ovarian follicles in mRNA expressions, and beta-actin gene was used as a control gene just to help us decide whether the gene of interest was expressed well or not. The findings of the study imply that GDF9 gene may play an important role in the growth and development of the ovarian follicles of Potchefstroom Kokok chicken breed. The paper published in 2018 have found that the expression of GDF9 mRNA is high in small yellow follicles when compared to larger yellow follicles in these mentioned breeds, and that may be due to breed differences. These results agree with the previous studies published in 2018 that GDF9 gene might be a molecular marker for improvement of egg production. The study concludes that GDF9 mRNA expression was found in all the follicles from follicle 4 to follicle 1, with high expression in follicle 4 and follicle 1. This indicates that GDF9 gene is playing an important role in sustaining the growth and development of the ovarian follicles of Potchefstroom Kokok chicken breed. It is recommended that further studies be conducted on the expression of GDF9 gene on some other South African indigenous chicken breeds and in other egg production related genes in Potchefstroom Kokok chicken breed. The findings of this study forms part of the work that was published in an accredited journal, which is Brazilian Journal of Poultry Science. These are some of the references that were used to compile the presentation. I would love to acknowledge the University of Limpopo, Department of Agriculture, Economics and Animal Production for giving me an opportunity to perform the study. I would like to give thanks to my supervisor, Prof. Jasi, my co-supervisor, Prof. Mbazima, for the guidance and support. I'd also like to thank JS research team for building me and giving me the confidence. Ladies and gentlemen, this marks the end of my presentation. Thank you.
Ladies and gentlemen, we heard the presentation from Victoria Tlokwe. This is the world of proteomics and genomics. I believe she spared us more than 5 minutes for the questions. Right. Do we have people who want some clarification? Our revolving mics, please come forward. We have one here. Do you have anyone else? Just take note that we have more than 5 minutes. We have another one there. Okay, you can go ahead.
Okay, thank you very much. Thanks for the presentation, concise and straight to the point. Okay, I just want to find out from your design. You spoke about, you used random design. I'm not quite sure what does it mean in terms of the setup that you have, that you had.
Yes, should we have the microphone at the back there? There's a hand. Is there a technical problem there?
No, it's just a bit of a confusion. Thank you for an interesting presentation. I don't know if I missed it. I just wanted to know what was the differences between the follicles Was it size or what was the difference? Thank you.
Okay, Ms. Tlokwe, you have 2 questions, and I am glad that I can add 1 to make them 3. In your post-hoc test, the mean separation, you used the Duncan multiple range test. Amongst the host of the post-hoc tests, probably there are about 7 of them. Why specifically that one, the one that you used?
Okay, thank you for the questions. And for correction, the design that I used is completely randomized design. And this design is used when you have uniform experimental designs. And in my case, that's what was happening. And then all the chickens that were used, they had equal chances of being selected for the purpose of the study. And to answer the second questions of what was the differences between the follicles, yes, is the size. The follicles differs in size, and we have many follicles. And in my study, I selected the ones from F4 to F1. And then the difference is just their sizes, the diameters. Okay, and then the last question of why Duncan post-hoc. The reason why I chose to use, Duncan multiple range test is, yes, there are many post-hoc tests, but according to my understanding, they just used to find the differences in the means. So I just chose to use Duncan as one of those. So yes, I understand that they just perform the same thing of checking the differences. Thank you.
Thank you for the responses. I believe we can take only another set of questions.
Just a quick one for someone who's not in your field. Overall, what does your work and results mean to a farmer who's farming with Potchefstroom kukuk?
Okay, thank you for the question. In this study, the main thing was to determine the expression of this GDF9 gene that was found by the scientists to be playing a role in the production of the eggs, especially the development of follicles. So what happens is that GDF9 helps us to, the gene expression, helps us to find or identify the genes that might play a role in the production of a certain product, which is in our case is egg production. So, in my study, I found that the GDF9 is expressed high in 2 follicles, F4 and F1, and it was expressed low in the 2 follicles. So what can be advised is that, since it is expressed low in the 2 follicles, now it raises a concern to say, maybe the reason why this chicken is not doing so well in egg production might be because of the low expression of the gene. So now to the farmers, it can be that when now they want to genetically improve the egg production of this chicken breed, since now there are some markers which were found as in the gene expression levels, which shows that the egg production might be improved when you improve the presence of the gene in those follicles. So we might say that they can sell, when they are doing selection during breeding, they can breed those ones which are having enough expression or high expression, with a hope that they will get an outcome that is having, the chickens that are having more production of eggs. And that will benefit them a lot, because now they will be able to make more profit in their cells with more production. Thank you.
Thank you, ma'am. Due to the issue of time now, I think we can release the presenter. We move on to our next presenter. Is Portia around? Portia? Portia Morifi. We're going to listen to the presentation regarding sweet potatoes.
Greetings, everyone. I hope that everyone can see me because it's clear that God, it's clear that God has His own favorites when He was giving out the heights. Standing before you is Portia Mrifi, and I'm here to present my research findings of the research titled Host Status of 19 Seed Potato Lines to Milledgine Incognita and Milledgine javanica, compiled together by my co-authors Bofu and Mashela. Sweet potato, which is scientifically known as Apomia patata, serve a significant role in promoting food security, human nutrition, and poverty reduction. Most crop-producing regions are replete with root node nematodes that causes a major production loss in the agricultural production while starting from the small scale to commercial farmers. The previously widely used fumigate nematicides were used to manage high nematode population densities were withdrawn from the agricultural markets due to their negative impact on the environment, humans, and animals, including soil deterioration and ozone layer depletion. And therefore, the use of resistant plant genotypes serve as an alternative nematode management strategy. Figure 1 shows a highly infested sweet potato tuber showing the galls and the crackings caused by the nematodes. Milledgine incognita and Milledgine javanica are widely distributed in sweet potato producing regions and they mostly occur as mixed populations. In nematode susceptible sweet potato cultivar, both yield and market value of produce are negatively affected as the nematodes cause the root galls on the plants that reduces water absorption and nutrient intake. The use of nematode resistant in managing nematode population densities requires sufficient knowledge on both nematode species and host plants. Historically, the two Milledgine species were viewed as being widely distributed, and the Agricultural Research Council sweet potato lines are being bred for their high nutritional values through the process of biotification. And among the 19 sweet potato lines, 5 of the lines were tested resistant against Forasium, and the reproductive potential is one of the descriptors available to assess the host status. The aim of the study was establishment of the host status in 19 ARC sweet potato lines to 2 Milledgine species, and the objective of the study was to determine whether the reproductive values of the 2 Milledgine incognita on all 19 ARC sweet potato lines will provide evidence of non-host, and the null hypothesis was therefore derived from the above objective. At the Green Biotechnologies Research Center of Excellence, University of Limpopo, 2 parallel studies for Milledgine incognita and Milledgine javanica were conducted under greenhouse conditions, and 2 trials containing Bio45 19 seed potato cuttings, including Bericide serving as a susceptible cultivar, were arranged in a randomized complete blood design with 6 replications. 20-centimeter plastic pots were filled with growing mixture of steam pasteurized loam soil, sand, and hygromix at a ratio of 3:1:1, and the spacing for pods were 0.25 centimeter by 0.30 centimeter intra-row spacing. And the inocular was placed in 5 centimeter deep soil on cardinal points of vines with hole covered using the growing mixture. And the inocular was prepared by extracting the nematodes from a highly susceptible tomato cultivar Floridate. And 7 days after setting, the cuttings were inoculated with 250 eggs and second stage geolan of each melanogain species using a 200 milliliter plastic syringe. At 56 days after inoculation, the fresh roots were then collected and root culling was rated using a North Carolina differential scale of 0 to 5. The eggs and J2 were extracted from roots through maceration and blending for 30 seconds in 1% sodium hypochlorite, and then later counted from a 5 milliliter aliquot under a stereo microscope at a magnification of 60. The reproductive potential data were subjected to analysis of variance using statistic 10.0 for each trial, and the reproductive potential was then computed for each line using the X plus second stage juvenilines over 1 gram of fruit. Figure 2 shows the trials for the Melanogain incognita species under greenhouse conditions. And figure 3 shows sweet potato roots inoculators with Melanogain incognita before the root gall rating. Table 1 shows the partitioning mean summer squares into total treatment variation of reproductive potential of Melanogain incognita and Melanogain javanica. And the treatment effects on the reproductive potential contributed 40% under Melanogain incognita and 38% under Melanogain javanica. Table 2 shows the reproductive potential of Melanogain incognita and Melanogain javanica on sweet potato at 56 days after inoculation. The reproductive potential is determined when the RP, and when the RP is less than 1, it suggests that the tested sweet potato is a non-host to the tested Melanogain species, and when greater than 1, it means that the tested sweet potato is a host to the Melanogain species. Looking at the reproductive potential of both the Melanogain species, starting with the Melanogain incognita, it shows that Melanogain incognita failed to reproduce only on 4 lines, while Melanogain javanica failed to reproduce only on 2 lines. Internationally, Melanogain incognita is viewed as being more aggressive than Melanogain javanica, but vice versa in South Africa. And the result proves that Melanogain incognita is indeed aggressive than Melanogain javanica in South Africa, as Melanogain javanica failed to reproduce on only 2 sweet potato lines, and Melanogain incognita failed to reproduce on only 4 lines. Among the 15 lines, there is 5 lines that are resistant to Fusarium and Melanogain javanica infested on all of the lines, while under Melanogain incognita, only 2 lines were non-host to the species, and cultivar Beregat served as the susceptible cultivar. The null hypothesis was then rejected due to the existence of non-host status among the 19 ARC sweet potato lines based on their sweet potato lines based on their reproductive values below unity. We then recommend that further investigation on relative susceptibility and mechanism of nematode resistance involved is necessary to establish whether the sweet potato lines could be used in integration during the plant breeding, which are my objective 2 and objective 3 of my dissertation. Listed are all the references used to compile the presentation. I would like to thank the University of Limpopo for enrollment, the Agricultural Research Council for consumables, and the National Research Foundation for funding. Thank you.
She was just on time, so we have plenty of time for the questions. Prof, here, do you have somebody else?
Okay, thank you very much for your nice presentation. I think in your recommendation, you made a recommendation saying that further investigation should be made. Who is going to do that investigation? I think it's a general recommendation. Can you tell us who is going to do that investigation? Which
Okay.
department and so on? Thank you.
Can I answer? Okay, Prof. I have mentioned that those are part of my objective in my dissertation. So I'm the one who's going to investigate.
You have already done the study. You mean, are you planning to do, is it a PhD or it's a master's, isn't it?
It's a master's.
So you are planning to do your PhD and then go ahead and start investigating?
No, they are part of my master's objective, which is objective 2 and objective 3.
Yeah, but the recommendation that you made, further investigation is required. Who is going to do that further investigation? That's the question that I asked. Are you going to do it yourself?
Yes.
Oh, that's good.
Thank you, Portia, for the presentation and response to the questions. You can now be excused. Our next presenter, ladies and gentlemen, is Rachwene Pretty. She's going to present on seasonal changes in the nutritive value of forage grasses, an experiment that was done at the University of Limpopo Experimental Farm.
Good day. My name is Ratweni Prete, and I'm a student at the University of Limpopo doing masters. And I'm under the supervision of Dr. Mukwabana, Dr. Nemutandani, and Professor Jasi. Today I'll be presenting on seasonal changes in nutritive value of some identified grass species at Seyferkel University Experimental Farm. This is the presentation outline for the study. The nutritional quality of the grasses is affected by abiotic and biotic environmental factors, which includes the soil type, the climatic regions, and even the management practices. The rangelands cover more than 80% of South Africa's land surface, and it is critical for sustained provision of essential ecosystems of goods and services, such as feeding for livestock. They also make an obvious contribution to the development of the economic value of the country through agriculture and supply of affordable food products. Figure 1 shows grassland. If you look clearly, you can see that the grass that is there in the middle is Heteropogus conctortus, which is one of the species which were identified at the farm. The diet of ruminant animals in South Africa is mainly comprised of the natural pastures. As you know, there's a saying that says, grass is the cheapest source of feed. If you were taught by Dr. Muteka, you probably know this song. The poor nutritional value of natural pastures, they reduce the performances in production of their livestock species in terms of maintenance, the growth rate, milk production, and others. As a result, farmers resort to purchasing additional feed to supplement poor quality pastures, and this increases the cost of production. The grass species are also considered as the main plant type of forage, which plays an important role in feeding of the livestock for arid and semi-arid regions. And however, the nutritional compositions of these grasses are affected by the seasonal changes. According to a study conducted in 2004, it was reported that during the dry season, rangeland grasses have low crude protein and fiber content. Pastures grow rapidly during the rainy season, and the nutritional value is high, as stipulated by the study conducted in 2007. Multiple studies have spoken about the nutritional values and how it's affected by seasons in many other areas. However, information on the effect of seasonal changes on nutritive value of some identified species at Fertility University Experimental Farm is not yet known. Therefore, the aim of the current study was to evaluate the nutritive value of these identified grasses at the farm during the dry and the rainy seasons, and the objectives were to identify the grass species available there in these two seasons, and to also determine the effect of seasonal changes on the nutritive composition of these grasses during those seasons. And the hypotheses are in line with the objectives. This study was conducted at University of Limpopo Fertility Experimental Farm, which is situated 9 kilometers northwest of the University of Limpopo Tafelop campus. A systematic quadrant sampling method was used for the study, whereby a quadrant was thrown every 2 meters, and the grass species within were identified using the identification guide by a study conducted in 2015. The grass species were collected at the farm and were analyzed at the University of Limpopo Animal Production Laboratory for dry matter, crude protein, moisture content, ash, and gross energy. All these analyses were repeated 3 times. Data collected was analyzed using statistical analysis system version 9.4, and analysis of variance was used to obtain the objective at P is equals to 0.05. And the following statistical model was used in the analysis. Table 1 shows the results on the identified grasses in both seasons. There are 8 in total, and it also shows the common names. If you look at Table 2, which shows the nutritive value of the grass species during both seasons, for example, if we look at the moisture content, we can see that there was a significant difference in the performance of most of these grass species except Eragostris rigidia and Temeda trianda. If you look at the one highlighted in greens, you can see that there was a significant difference in those ones. Similarly, for gross energy, which is the last column there, you can see that the rainy season performed much more better than the dry season. The results of the current study show that there was a significant difference in, for example, the crude protein of Hyperthelia disolute, Malinus repens, Temeda trianda, and Schmiditia perforates, with the rainy seasons performing relatively better as compared to the dry season. This implied that the aforementioned species can be used for feeding to account for the protein requirements of livestock, thus to improve production performance. The findings are supported by a study conducted in 2004 who reported low crude protein of grasses in the dry season. Gross energy also was high in all grass species in the rainy season, meaning that these species are rich in total digestible nutrients. A study conducted in 2008 also reported higher gross energy values in the rainy season. The study concludes that the rainy season was high in moisture content of identified grass species except Eragostris rigidia and Temeda trianda. This means that all those grass species that performed better in this moisture content, they can be used to promote moisture in feeding of livestock. The overall performance in the nutritive values was high in the rainy season than the dry season, which means that if you want to account for nutritional requirements in livestock, you can use grazing as part of feeding in that season. Grass species such as Temeda trianda, Heteropogon contortus, and Malinus repens can be used to improve production performance in livestock, as these species are rich in crude protein and gross energy. The study further recommends that more studies should be done in a broader geographical area using more grass species and accounting for other nutritional components. I would like to acknowledge the University of Limpopo Department of Agricultural Economics and Animal Production, my co-author Majali Bodu and us, the supervisory team, and the JASI research team. These are some of the references I've used in compiling this presentation. Thank you.
Thank you, Rashene, for the presentation. You've spared yourself 5 minutes for the questions. Ladies and gentlemen, she is ready to respond to all the questions that you might have. Okay, you can go ahead.
Sure. Thanks, Pretty. It is known that the nutritive value of grasses will be higher in the wet than in the dry season. I just want to understand what were the reasoning for the study. I don't mean to corner you, just if you can't answer, say so, it's not a problem. What's the other issue? What may be of importance to your work, if maybe it's not considered, is that farmers often don't know exactly when to start supplementing. So this could help if you determine in your study area that at this particular time, you know, the nutritive value of this dominant grass species is going down, and therefore farmers could, you know, start. So something along those lines that you may want to consider. And the other thing is, and that would mean that you need to not broadly look at the dry and wet, but early, mid, and late, both dry and wet season, so that you know exactly at what stage. The other point that I will suggest, I wonder whether if you also know or have looked at the field management regimes that may have influenced the nutritive value. For instance, fire as one, and grazing intensity. I'm saying that because Melanis repens, it's often found in disturbed area, and so that could be as a result of an area that often bends or that is hammered or overgrazed. And did you also sample your plants at the same time in the wet and the dry season?
Thank you for those wonderful questions. In a few seconds, she will be up here responding.
I do understand. Thank you very much for the questions. In terms of the other two suggestions you've made, I'll note them as comment and I'll consider them further. The sampling, no, it was not done at the same time. I think it was because of the period that was given to conduct the study, since it was conducted in 1 year. So it wasn't necessarily sampled at the same time. In terms of the field management regions and these ones, I could also suggest and recommend that further studies could do studies related to that one, because this is a limitation in my study. The significance of the study is, in my conclusion, remember that I told you that the nutritive values of these species, I mentioned the direct species, for example, that the nutritive value of species like tomato trianda had high crude protein. So when I was concluding, I was also suggesting which species can be used to improve production. I was not just talking about the season alone. So the significance of the study is that farmers can use these species that I have recommended in this study to improve production in their animals. Thank you.
Thank you, Pretty, for the responses to the questions and your presentation. In the absence of difficult questions, you are excused. Our next presenter is Ruben Mohalo. Can you approach the podium? He is going to talk about the ICT tools that are being used by the small-scale farmers in Blueberg local municipality.
Good morning, everyone. Go by the name Mongalu Ruben, and I'm here to present about the title of my study is investigating adoption of information communication technology (ICT) tools by small-scale farmers in Blueberg local municipality, supervised by Prof. Zwane and Mr. Mpatlele. Here, like is my outline, as the outline of my study. With the agriculture plays a crucial role in ensuring global food security and economic stability. As of that, small-scale farmers play a vital component of agricultural production in many regions, as they also contribute to the livelihoods of rural people, and also to contribute to the economy of the country, and also to food security as well. Yet small-scale farmers face various challenges, including limited access to resources. For example, the use of production inputs. ICT tools encompasses of wide range of digital technologies, including mobile phones, internet applications, sensors, data analytics, and clouding as well. The problem statement: extension officers are the main current source of information. The adoption rate among small-scale farmers remain a subject of concern, as how the levels and the rates of ICT tools they have adopted or rejected small-scale farmers. Some of the reasons include low levels of education, adoption of technology, high cost of broadcast equipment, and age as a factor. These challenges simply emphasize the need to use hybrid methods by agricultural extension service delivery. In South Africa, availability of electricity is a major concern for small-scale farmers to adopt ICT tools. The motivation of the study is that small-scale farmers in South Africa could benefit more from the use of ICT tool access to agricultural information, but yet, to the extent of the adoption of ICT tools by small-scale farmers is currently unknown and undocumented. The study offers to bring to light the adoption of those ICT tools by small-scale farmers in Blueberg local municipality. The research seeks to address the following key questions. What is the current level of ICT tool adoption among small-scale farmers in Bloberg local municipality? And also, what are the primary drivers and barriers influencing small-scale farmers' decisions to adopt or reject ICT tools for agricultural purposes? The last one, how does the adoption of ICT tools impact small-scale practices, productivity, and overall livelihood? The studies aim to investigate the adoption of information communication tools that can be used or adopted by small-scale farmers in Bloberg municipality. The objectives of study are as follows: to identify the current level of ICT tool adoption among small-scale farmers in Bloberg local municipality, to identify the drivers and barriers are influencing small-scale farmers' decisions to adopt or reject tools for agricultural purposes. The last one is to investigate the adoption of ICT tools' impact on small-scale farming practices and productivity and overall livelihood. With the methodology, the study for that is still ongoing study. So the research is using mixed approach methods, the use of qualitative and quantitative data. For that also the researcher used a pilot study to test the reliability and the validity of the research instrument, which is a well-structured questionnaire. The population of the study included 6 villages, which is Tebosgrot, Kromhoek, Difred, Grootpan, Eldorado, and Haramasuga. The estimated number of farmers is 210, which constituted the research population. The study uses a random sampling for selecting. Also, the researcher selected 35 farmers per village for participating randomly. So the data is going to be analyzed using SPSS, Microsoft Excel, descriptive statistics. The data from pilot study was analyzed using Microsoft Excel. These are the results from the pilot study. The results shows the fields owned by small-scale farmers. The blue one, with the blue ones is the frequencies, and the orange ones like the percentage. It showed the results showed that mostly farmers owned phones as compared to other tools like radios and TVs and computers, as well as clouding and other things. So at the following the figure also it's showing the factors that are faced by farmers using ICT tools. Number one being network coverage. Due to load shedding in the country, they have highlighted that during load shedding, most of them they don't have enough load shedding, poor infrastructure in the country. And also the second one was financial support. They have highlighted that mostly they are not getting any financial support from Department of Agriculture due to certain policies that limit them, because they don't have some of the things. Mostly they were talking about water use authorization, meaning that to get that certificate is expensive for them, so it's a kind of a hinder for them also. And also with this thing of education, most farmers they're not all literate. So for them to use those ICT tools, mostly navigating through the clouding, more of a survey 123, where it's a reporting tool for extension officers, but also when the extension officers go to the farmers, they also teach them how to use it. But in return, it's a tool for extension officers to report back to the stakeholders, mostly back to the department of the findings that they found there, farmers that are experiencing. With the last one is a conclusion and recommendation. The key challenges to increasing the use and adoption services in developing countries is the lack of access to affordable and accessible information and communication technologies. So with this, mostly I suggest that stakeholders invest more in ICT for small-scale farmers to be efficient and effective in contributing to the food security and economic stability of the country. And governments and stakeholders play important role in promoting use of adoption of information services by farmers. This can be done by pushing extension officers to promote ICT tools when they're organizing farmers events, farmers market and our information awareness. Teach farmers about the ICT programs, the adoption and the use, so that farmers, we're moving into the fourth industrial revolution where farmers must understand the use of technology and how to operate it for the future reference and the betterment of the country. I'd like to acknowledge AgriSeta for funding Prof. Zwane and Mr. Mpatlele. Below like the references of this presentation. Thank you.
Thank you, Ruben, for the powerful presentation on ICT. Ladies and gentlemen, we heard the presentation. Do we have questions for the presenter? Questions for the presenter? Okay, it means that it's your lucky day and you were audible enough for everything. Oh, wait. Okay, you can go ahead.
That will be unfair to others. Let me see. You mentioned something about water license and the fact that one of the constraints to farmers was the support from from the government. How was that has to do with ICT adoption? And I know that when you're talking to farmers about a particular aspect, they will tell you about a whole range of other issues. And one of the problems that I've also highlighted, I know about, is the dependency. And I don't mean to be harsh from our farmers, but there is that element. What's the other question that I had? Okay, the other one is just a comment. The likes of the ARC ICT has a number of apps, for instance, and but I'll talk to Prof. Zwane, which could then link well with your work. Just answer the one question.
With the one of water use authorization, is linking back to the funding, where like most of the farmers, like with ICT, the adoption, it makes it difficult for the farmers to adopt that due to the fact of affordability. If they can get the access to funds by the Department of Agriculture, with like mostly if like there's a program where they're saying like it's a farmer support program, and they're asking for water use authorization rights, which is a limiting factor to the farmers, like to apply for that program, to access the funds, also to buy those ICT tools. It's a limiting factor to that.
Okay, we are just waiting for a confirmation if your response was satisfactory. Okay, it's confirmed. Now you can. Colleagues, I just want to confirm if the following two presenters are here. We have Taviso Shosi and Shekofajo Shai. Are they here? Oh, that's Shekofajo, right? Taviso. Okay. Now our next presenter is Sharon Lovu. Sharon, please, you may approach the podium and give us a presentation on the indigenous crops in mineral contaminated areas. you upload your presentation? Okay.
Okay, I go by the name of Ndlovu SN, and I'm here to present my study, which is titled Exploring the Use of Indigenous Crops for Heavy Metal Phytoremediation of Mine-Contaminated Soils. And I'm under the supervision of Dr. P.M. Hopa and co-supervised by Dr. M.J. Mutapo. So this is the content of my presentation. So mining has resulted in a large number of environmental pollution, including soil contamination, and thus causing problems to soil health. So heavy metals are one of the commanding contaminants in the environment, and especially in areas which are surrounded by mines. So as a result of these contaminants in the soil, approaches such as phytoremediation have been developed, which will reduce the heavy metal contaminations in the soil. So phytoremediation is regarded as a cost-effective method of utilizing crops to remediate contaminated sites. So in the available literature, there is limited information on how indigenous crops can be used in the remediation process. And thus, there is a need to explore how indigenous crops can be used to phytoextract and reduce heavy metal concentrations in mine-contaminated sites. So this study aimed to investigate the potential of selected agricultural indigenous crops for phytoremediation of mine-contaminated soils. And the objective was to determine whether the selected indigenous crops will decrease heavy metal concentration in mine-polluted soils through phytoremediation. So the study was conducted in two areas. The first area was where the soils were sampled, which is in Tikontele Cooperatives Farm, which is in Barberton, Mpumalanga, having a mean annual temperature and mean annual precipitation as stipulated. And then the pot study was conducted at the Green Biotechnology Research Center at the University of Limpopo. So figure 3, as displayed, shows the location of where the soils were sampled in Mpumalanga, which is in Barberton, and the farm is next to a mine which is called Lily Mine, which is a gold mine in Mpumalanga. And the farm, which is called Tikontele Cooperatives Farm, is a community farm which produces sugar cane, which is then later produced to which makes sugar. And then the farm is divided into about 7 fields, but then this study only was done in field number 1. So, so the experiment comprised of 5 treatments which were arranged in a randomized complete block design, replicated 5 times. And the treatments comprised of crop species Amaranthus hybridus, Cleum gynandra, Bidens palosa, Cucumis africanus, and a control which just contained soil without any crop species. So the seeds were then planted into the pots containing 3 kg of soil each, and were placed under greenhouse conditions. And then they were then irrigated with deionized water to avoid further contaminations from the water. So the experiment was terminated when each crop species reached flowering stage. So data collected included soil pH both in KCl and water using the electrometric method, soil electrical conductivity using the EC meter, and heavy metals from the soils were extracted using the EPA method, which uses the ICP OES model. So all data was subjected to analysis of variance using GenStat version 18 by a one-way ANOVA at p less or equals to 0.05, and the means were separated using the Duncan's multiple range test to test for any significant differences. Then under results and discussions, so table 1 shows the response of non-heavy metals in mine-contaminated salt after the phytoremediation process. But then under the treatments, we have treatment number 1, which is PP, which shows the soil or the heavy metals in the soil before phytoremediation, or shows the concentration of the heavy metals in the soil before phytoremediation. And then as shown in the table, the treatments were highly significant with arsenic, cadmium, and also lead. And then amaranthus was a high reducer of heavy metals, arsenic, cadmium, and lead, while Clium gynandra was a high reducer of heavy metal chromium. And then the control treatment was then also reduced a little bit of heavy metals. And this shows that even though the control managed to reduce heavy metals, but then it was not as effective to have those crops. So the relative impact of arsenic, cadmium, and lead are, as shown in the table, on amaranthus are 41.49%, and 50.90, and also 51.74 respectively, which shows that amaranthus was the high or most effective crop. And also in generally, I can say amaranthus was a good remediator for most of the metals. Then table 2 shows the permissible heavy metals in the soil as outlined by FAO and WHO. And according to the table, or according to the results, all the other heavy metals were below the permissible. It's only arsenic which was above permissible after phytoremediation. And this is a problem because arsenic in the soil arsenic is a toxic and a non-essential heavy metal, and if it is in the soil, it will affect the root of the plant, and also it will then later affect the growth of the plant. So, and the reason maybe the heavy metals in the soil, like arsenic in the soil, was more than the permissible is because this phytoremediation process was done for only 8 weeks, which is up to flowering stage of the crops. And if maybe it was there for longer periods, more heavy metals would have been extracted. So, chemical properties also like pH and EC were done in the soil. So before phytoremediation, or the original sample had a pH of 7.45 in water, and in KCl the pH was 5.45, and then the EC was 84.8 microsiemens per centimeter. And then table 3 just shows the concentrations of the pH and EC after phytoremediation with the different crops, and also in the control sample. So and as it is shown in the table, the parameters all increased in the concentration, and this is a problem, especially in pH, because it is a driver, I can say, or it blocks the availability of other or most nutrients and minerals. So the findings of the current study are in line with those by the mentioned author, who also observed an increase in soil pH after phytoremediation. And also an increase in the pH may be due to cation and anion imbalance in the soil, or the release of organic anions. And also, according to the author, EC is classified into 3 groups or 3 categories, and based on the results of the current study, the soils are classified to be non-saline because the concentration is less than 200 microsiemens per centimeter after phytoremediation. So in conclusion and recommendation, the different indigenous crops in the current study succeeded at reducing the heavy metals in the contaminated soils. And this shows that the indigenous crops that were used to remediate the mine contaminated soils support the phytoremediation process. And this was evident for the heavy metals, arsenic, cadmium, chromium, and lead. And also phytoremediation with the different indigenous crops increased the pH both in KCl and water, and also the electrical conductivity. So in recommendation, I would say that there is a need to look into other elements since the pH increased after the phytoremediation process. And also, I will recommend that the crops in the soil should be in the soil for longer periods for optimum removal of heavy metals. So these are the references that I used, and I would like to acknowledge my supervisors, Dr. P.M. Hoba and Dr. M.J. Mutapo, for their support during the study, and I would like to acknowledge Deacon Cooperative Farms in Barberton, which allowed me to do my research at their farm, and I would like to acknowledge the University of Limpopo for giving me the opportunity to do my study. And I would like to thank each and every one of you who's here, who listened to me. Thank you very much.
Thank you, Sharon, for the presentation. I can see that the people holding the revolving mic are ready. Do we have questions? Do we have questions? Abigail, behind you, we have one. And we have another one at the back. Then, when you are done there, we have another one in the front. It seems as if we have a couple of hands here. So we will try to limit our questions to only one per person.
Okay, thank you for your presentation. I just want to know what criteria was used in choosing this different plants, because we know that most plants can uptake trace metals from soils.
The second question? Yes, you can go ahead.
Nice presentation, very interesting topic. My question is on the crops that you are using. So what will happen now to the arsenic that has been absorbed by these plants, the plants? How are you going to, maybe when it coming to, maybe when you cut off the plants, what will happen to the heavy metals that you've removed from the soil, but they are in this plant?
Okay, the third question?
Mine is very simple. You said that you irrigated your pot using deionized water to avoid further contamination. Why not distilled water?
Okay.
Yeah, thank you so much. My question is, what is an issue? What motivated you to look at heavy metal contamination in the soil? In other words, what's the major driver? What really causes contamination of soil with heavy metals? Just want to understand the motivation of you looking at heavy metal contamination in soils. Thank you.
Okay, thank you very much for the questions. The first one, I think if I got it correctly, was what was the criteria that was used in choosing the crops that I choose. So the criteria that I used was just, I checked indigenous crops that I can easily find around, which I can use during the study, and those crops which are able to grow also in conditions in the soil which has heavy metals, and also those crops which are able to take up those heavy metals or are able to phytoremediate heavy metals in the soils. So that's the criteria that I used. And then the second one is, I feel like it's in line with the first one, to say after the heavy metals or the crops have taken up the heavy metals, what is going to happen? So the crops that are used, like I'm saying, they are able to grow even though they have those heavy metals in them. So nothing like is going to happen to the crops. The heavy metals are not going to damage the growth of the crops. And then the third one was why use distilled water instead of, or why use the ionized water instead of distilled water? So I don't really have an answer for this one because when I requested for either of the two water, I was told that the ionized water is the one which is available, and it is also purified, doesn't have any contaminants. So yeah. And then the other one was, what was the motivation maybe of the study, I can say, which drives me to do the study. Okay, so what drives me to do the study is the farm in Barberton, plants sugar cane, as I said, and after observing their sugar cane, the growth was not uniform. So, and also since the farm is next to the mine, I also observed some deposits from the mine in the soil, which shows that they are from they are from the mine. So hence hence the motivation to do this study. Thank you.
Thanks, Sharon. Just a quick one. You have some very interesting results. Did I hear you correct that your control, particularly I think on lead and the other one, also, reduced or absorbed, some of these metals? And I wonder why, because there was nothing planted. And just a quick one is, now that you have seen that some of these indigenous plants can absorb, what's next? Are you going to tell the farmer or the mine to plant this, and for what reason beyond just absorbing them heavy metals?
Okay, thank you for the question. So after I've seen that the study was a success, the plan is to go back to the mine and tell them that, okay, this is what I've discovered in your soil. And the solution to that problem is this one. You can plant crops around the farm, not really like plant the crops all over. They can still also plant their sugar cane, but also have those crops, those indigenous crops in the farm, so that they can help take out the heavy metals which are damaging their sugar cane. Thank you.
Thank you, Sharon, for the bravery that you have shown in this presentation and response to the questions. You are excused. Ladies and gentlemen, can we applaud the presenter? From this moment, I would allow Dr. Nengovela to take over.
Good afternoon, ladies and gentlemen. I am Ruzani Nengovela, and thank you, Prof. Tada. I'm just going to continue from where Prof. Tada left off. Our next presenter is Relebohile Shibure, who is a master's candidate. Relebohile, are you here? Just to remind the presenters, you have 10 minutes for presentation and 5 minutes for question. The stage is all yours.
Good afternoon, ladies and gentlemen. I'm Shiburere Leborile, and I'm here to present my study titled Post-Harvest Physical Quality Changes of Cowpea Leaves Harvested at 2 Maturity Stages under the supervision of Satere TK and Mafio TP. The leaves of cowpea are one of the recommended indigenous vegetables in Africa that has a potential to reduce malnutrition in sub-Saharan Africa. The leaves are rich in nutrients such as vitamins, mineral proteins, and carbohydrates. They are also known for their antioxidant properties, which are human health improving factors. However, cowpea leaves are very perishable due to increased moisture loss and wilting, resulting in post-harvest loss during storage, leading to reduced shelf life. The shelf life and quality of cowpea leaves, the shelf life and quality of cowpea leaves are reduced within 24 hours after harvest, and this is attributed to maturity stage. A maturity stage is an essential pre-harvest factor that determines the storage potential and final quality of the leaves. For instance, a leaves harvested too early in the season or before optimum maturity are small in size and are more susceptible to wilting within 24 hours after harvest. On the other hand, the leaves that are harvested later are over mature with some signs of yellowing and are prone to moisture loss. Therefore, harvesting cowpea leaves at different maturity stages will indicate which stage is more susceptible to moisture loss and wilting, and which will be important for post-harvest management of cowpea leaves. Previous study has shown that maturity stage has an influence on post-harvest behavior, quality, as well as shelf life of leafy vegetables such as lettuce and spinach. However, information on harvest maturity on cowpea leaves is limited. Therefore, the present study was aimed at evaluating the effect of maturity stage on cowpea leaves. We hypothesized that cowpea leaves harvested at a later stage, which is 90 days after sowing, respire quickly and lead to reduced shelf life as well as poor visual quality. A sowing of cowpea was done at the University of Limpopo Aquaculture Research Unit. The growing media was pasteurized sandy soil and pen bark. The leaves were harvested at 60 and 90 days after sowing and transported to plant production laboratory immediately after that. The leaves of the same color, size, and with no signs of wilting were selected for analysis. Here is a picture of leaves harvested at 60 and 90 days after harvest. At 60 days, the leaves were at vegetative stage, whereas 90 days, the leaves were at reproductive phase, with about 80% of the plants having flowers. The leaves were stored at ambient storage, and during the shelf life, color, mass loss, wilting, and senescence were evaluated. Statistical analysis was carried out using a GenStat software. Data was analyzed using analysis of variance, and Duncan multiple range test was used for mean separation at 5% level of significance. Cowpea leaves harvested at 60 and 90 days after sowing did not differ in color in terms of A at harvest. However, there was a significant difference whereby the leaves harvested at 60 days after sowing had lower A value compared to 90 days harvest. The same trend was observed for B value, whereby the leaves at harvest did not differ. However, the difference was observed during storage, whereby leaves harvested 60 days after sowing had a higher B value compared to 90 days. This indicates that during storage, there was a decrease in the degree of greenness due to degradation of chlorophyll content. The leaves harvested at 60 days had significantly higher hue angle compared to those harvested at 90 days after sowing, indicating the yellowing of the leaves. Here is a picture of the equipment we used to acquire the A, B, and the hue angle. The leaves harvested at 90 days had significantly higher mass loss compared to those harvested at 60 days. This indicates that the rate of respiration at 90 days was higher compared to 60 days. The wilting of cowpea leaves was not affected by maturity stage. However, there was a gradual increase during storage. This indicates that the continuous increase in moisture loss resulted in the loss of, This indicates that there was a continuous increase in moisture loss due to increased respiration resulting in the wilting of the leaves. The same trend was observed for senescence, whereby maturity stage did not have any effect on the leaves harvested at 60 and 90 days. However, the significant difference was observed with storage duration. This indicates that the degradation of chlorophyll content resulted in the yellowing of the leaves. The purpose of the study was to determine the effect of harvest maturity stage on physical quality changes of cowpea leaves. The research result shown that maturity stage did not have effect on wilting and senescence. However, the quality was affected by storage duration. A mass loss was significantly higher in the leaves harvested 90 days compared to 60 days after sowing. The results suggest that higher moisture loss do not necessarily affect visual appearance of cowpea leaves. Therefore, our hypothesis is rejected. Thus, leaves could be harvested either 60 or 90 days after sowing. I would like to acknowledge the University of Limpopo as well as my supervisors. Thank you.
Thank you so much, Relebogile, for your presentation. The presentation is now open for questions, compliments, suggestions. Are there any questions for Relebogile? First hand there.
Thank you so much, Relebogile. Thank you for the beautiful presentation. I just want to know, maybe I have missed something. How many cowpea cultivars did you use for your study? And if indeed it was one, don't you think your null hypothesis could have been accepted, maybe if you could have used different cultivars?
Are there any other more questions? Okay, Relebogile, you can come and respond to the question.
Okay, thank you for the question. In terms of cultivar, I used one cultivar for the study, and then from that to, what was it? Did it have an effect?
Don't you think that you used one cultivar, your results could have been different if you considered different cultivars?
On that one, I'm not sure about the issue of cultivar, because I was only focusing on the maturity stage. So,
Are there any questions, more questions? All right, thank you so much, Relebogile. Can we please give her a round of applause? Our next presenter is Thabiso Choshi. Thabiso Choshi, are you here? Thabiso Choshi is a master's candidate. You only have 10 minutes for presentation and 5 minutes for question. Please make sure that you stick to the time.
Good day, ladies and gents. My name is Mr. Choshi Thabiso J. I'll be taking you through my research project titled Comparing Sentinel-2 MSI and Landsat-9 data in the detection of maize streak virus at different growth stages. This study was supervised by Professor Dau and co-supervised by Mr. F.M. Mashao. Maize, with a scientific name called Z-maize, is a stable cereal crop widely cultivated across diverse agroecological regions. It has become the lifeblood of millions, serving as a vital source of sustenance and economic livelihood. In the sub-Saharan Africa, maize holds an important position as the primary cereal crop, being cultivated across more than 25 million hectares, primarily by small-scale farmers. Maize accounts for about 15 to 50% of energy in the human diets in the sub-Saharan Africa, and also as a stable food for the majority of the African population. Maize is, however, threatened by various biotic and abiotic stressors, including the viral infections. Amongst these viral infections, the maize streak virus is a particularly destructive pathogen that can lead to a significant yield losses if not managed effectively. The virus belongs to the genus Mastervirus and the family Germanoviridae. It occurs throughout Africa, where it causes what is probably the most serial viral crop disease on the continent. Maize streak is acknowledged as the most commercially straining virus impacting the maize crop worldwide and in Africa. This disease is dependent on an insect vector called a leaf hopper. Sorry for that, it's not a grasshopper, it's a leaf hoppers belonging from the family Cacciidulaena. In South Africa, the virus is found to be the most widespread disease, with the KZN being one of the provinces most risk of getting the disease. So, with this problem, timely detection and monitoring of the virus at different growth stages of maize is essential for effective disease management and safeguarding maize yield. We all know that traditional methods are also being used to effectively manage the disease, but then they are found out to be labor-intensive, expensive, spatially limited, and may not provide timely information. This research aims to compare the performance of two satellite datasets in detecting the infections in maize and to determine their suitability for practical application in agriculture. Maize, as one of the major cereal crops, is widely used to produce human foods, livestock feeds, and industrial products such as cereals, the maize, cornstarch, and alcohol. The higher levels of infection can lead to substantial crop losses, affecting the livelihood of smallholder farmers and potentially causing food shortages, which will lead to price fluctuations in the local and the global and the local markets. Therefore, the effective management is vital to ensure food security and economic stability. Remote sensing technology offers a way to effectively detect the disease, particularly the satellite-based systems like Sentinel-2 and Landsat-9. They have become indispensable in the modern agriculture. Conducting a comparative analysis between the two datasets can help agricultural researchers and practitioners to understand which satellite is better suited for specific applications and growth stages. This is a typical picture of a maize crop that is infected by the virus. It is shown by the irregular streak, white or yellow colorations along the length of the leaves. The one on the right shows a healthy maize crop. Now, we all know that maize crops have various growth stages, but because of data limitations and accessibility, we've decided to use the most two distinct growth stages of maize, which is the vegetative stage and the reproductive stage. This study was executed at the Ofkolako farms in Zanin, Limpopo province in South Africa. The farms that are around Ofkolako have an average aerial coverage of 20 hectares. They are situated within the boundaries of Mopani district between the following coordinates. The farms are located at an elevation of 598 meters above sea level. The climate is categorized as mainly subtropical with hot and humid summers and mild winters, with the wettest months being from November to March. This picture shows the Ofkolako farms that were used to collect our data. The three red squares shows farms that were used for data collection. For field data collection, a field survey was conducted in March 2023, where purposive sampling strategy was used to collect the GPS points of healthy maize and for maize infected with the virus. Random sampling strategy was used to collect the GPS points of the surrounding land cover types. A handheld trimble GPS was the one that was used to collect the GPS points at a 3-meter accuracy. A total of 525 points were collected, which 75 points for each land cover. Out of the collected points, the collected points were randomly split into 70 points for training and 30% for validation. For remote sensing data, Sentinel-2 and Landsat-9 images were accessed from the Google Earth engine platform for both growth stages to test their sensors in detecting and mapping the virus. The satellite imageries for the vegetative stage were acquired on the 12th of March, and those of the reproductive stage were acquired on the 2nd of May. The images were then pre-processed in the built-in functions using Google Earth Engine, and we only used images with a less than 10% cloud cover for classification. The GPS points collected were then recorded in Excel as a CSV format and then imported into the Google Earth engines to overlay them with the images. Random forest algorithm was employed for the classification within the Google Earth platform. The reason for choosing for the random forest algorithm is that literature has shown that it has been successful in detecting maize streak virus, not only maize streak virus, but also other crop diseases. The confusion matrix was also generated for validation to calculate the overall accuracy, the producer and the user accuracy of our classifications. Results and discussion. The spectral response of MSV. The spectral response of MSV infected maize crop and the land cover types are shown below. The one on the left shows Sentinel-2 bands. The one on the right shows Landsat-9. On the Sentinel-2, the band blue and shortwave infrared one were successful in spectrally discriminating between the infected and the healthy maize, and on Landsat-9, only one band, which is the shortwave infrared 2, was successful. On the reproductive stage, only one band on the Sentinel-2 was able to discriminate between the two, and Landsat-9 was unable to discriminate between healthy and infected. The following graphs shows the producer and the user accuracy for both Landsat for both data sets and also on the both stages. These are the spatial distribution of the infected maize and the surrounding land cover types on the vegetative on Sentinel-2 and also on Sentinel-9. These ones are the spatial distribution for both data sets on reproductive stage, Sentinel-2 and Landsat-9. The following graphs, they show the aerial coverages of all the land cover types that are within our study areas. In conclusion, the result concurs with the result found by Dow, who found that Sentinel-2 research can discriminate the infected maize from healthy and the surrounding land cover types. It is also essential to acknowledge the challenges and limitations of the study. Factors such as cloud cover, atmospheric conditions, and the need for ground truth data for validation can affect the accuracy. Climate conditions, including temperature, precipitation, can also influence the distribution and severity. Integrating climate data with remote sensing data can provide a more comprehensive understanding of the MSV dynamics in maize field. That is a recommendation. A comparative analysis between the two at different growth stages offers valuable insight into the strengths and weaknesses of these satellite systems. The conclusion found out that Sentinel-2 outperformed Landsat-9 by 5% on the vegetative stage, and also on the reproductive stage, it also outperformed Landsat-9 by 5%. The findings of the study has demonstrated clear advantages of utilizing Sentinel-2 over Landsat-9 data. The research also highlighted the superiority of Sentinel-2, particularly looking at the higher spatial resolution, more frequent revisit time, and additional spectral bands, which makes it more effective tool for accurately identifying the areas infected by the MSV. The final resolution of this satellite enables the detection of small-scale infections, while the frequent revisit times provide real-time monitoring and timely responses to contain the virus's spread. Moreover, the advanced spectral capabilities enhance the sensitivity to early-stage infections, facilitating proactive disease management. Sentinel-2 emerged as a valuable asset in the early detection and management of the MSV, enabling better informed decisions to safeguard maize production. And for further research, UAV can be the one to fill in the gaps because of its higher spatial and spectral resolutions. These are some of the references that were used for the presentation. I would also like to acknowledge or give thanks to the Afkalaka Farms community for letting me do this research within their farms, my supervisors, Professor Dao and Mr. Mashao, and also NRF for funding this research. Thank you.
Thank you so much, Mr. Choshi, for your presentation. You were right on time, which means that we have 5 minutes for questions, discussions. Any questions for Mr. Choshi? We have one hand there. Any other questions? You can go ahead, sir.
Thank you for a wonderful presentation. My question is on your data acquisition. I see you used both Landsat and Sentinel. What was the reasoning for this, as they are both imagery, and you didn't mention the specific resolution of them? So what was the reasoning for using both of them?
Well, the major target of this research, was smallhold farms, but not just subsistence farmers, but also commercial. But then, because subsistence farmers lack capital, we then decided to use data source that is open open source. Sentinel-2 and Landsat-9 are freely accessible. That's why we used it. And also comparing it to other, yes, there are other data sets that are better than Sentinel-2 and Landsat-9, but those ones are commercial. You have to pay to get their data sets. So the reason we used these two is because of the cost, because we are also targeting small-scale farmers.
Thank you. There's a hand there at the back, and there's another one there. Yeah, you can. Go ahead, ma'am.
Thank you very much for a beautiful presentation. I would like to ask the size of the area where the maize was planted. How the size of it, can you please mention it?
Okay. Since I collected my points from 3 different farms, each farm was about 4 hectares, the small scale 4 hectares, but the results were also affecting the surrounding farms. So overall it was a 20-hectare study area, 20 hectares. But the data was collected from 3 farms.
Thank you, Mr. Joshi. It was a very good presentation. Mine is a comment. Your title is a bit misleading. You are using remote sensing to identify a virus. I would have preferred maize streak disease. So that's my comment. Thank you.
That was a suggestion. It was not a question. Let's give Mr. Joshi a round of applause. Thank you for your presentation. Our next presenter is Chirizi Mukwebo. Are you here? Chirizi Mukwebo is also a master's candidate. You have 10 minutes for presentation and 5 minutes for questions.
Good day, ladies and gentlemen. My name is Chirizi Mukwebo. I am a Master of Science student at the Department of Geography and Environmental Studies. I am here to present one of my research objectives titled Land Use and Land Cover Change Detection Using Remote Sensing Data in Panama Village, Sekhukhune District, South Africa, under the supervision of Professor Aidau and Professor K.K. Aisi. This study is part of a national project called the Global Environment Facility 7, in short is known as the GEF7, in efforts to achieve land degradation neutrality by 2030. This is in line with the Sustainable Development Goal 15, which serves to protect, restore, promote sustainable use of terrestrial ecosystem, sustainably manage forest, combat desertification, and halt and reserve land degradation and halt biodiversity loss. We have all here participated one way or another in the manipulation of land, whether knowingly in terms of you have learned about it and you are aware of your actions, or just doing it because it's part of life. I mean this by we live in houses, we have businesses, we have workplaces, we traveled from our homes to this venue, and we are in a venue. This is part of land use. The fact that there is a building here, this is what land use is. Before this building was here, there was just a natural land in this place. So that is what I'm going to talk about. Land use refers to the manner in which human made use of land and its associated environment, typically emphasizing the practical function of land for economic purposes and also for domestic purposes. And in this land use, we find land uses as such as overgrazing, such as built-up areas, and these, when we manipulate land like this, we change the land cover. Land cover refers to the physical attributes of the earth's surfaces, encompassing the arrangement of flora, the water bodies, the soil, and other physical elements of the land. Limpopo is one of the most severely degraded provinces in South Africa, as identified in the GF7. And Panama was one of the focal study areas that was identified as the most severely degraded village in Limpopo. Land uses such as overgrazing and unsustainable land use practices were found to be the primary causes of degradation in Panama village. This was accompanied by the social ills, the fact that they do not have water in the area, and that the village is impoverished was also a factor in this study. These causes were closely linked to population growth in terms of the poverty and also the land uses that are unsustainable in the village. The aim of the study was to detect and to map land use and land cover changes using geospatial technologies, and the objective was to map and quantify the land use and land cover changes in Panama village. The study area's climate is classified as hot, semi-arid, with summer temperatures ranging from 28 degrees Celsius to 17 degrees Celsius, and the winter high and low temperatures being 19 degrees Celsius and 8 degrees Celsius, with the annual average rainfall of 250 millimeters. We used secondary data for this study, and data was acquired in the website United States Geological Survey in the link that is provided there. We used Landsat 5 TM images for the years 2000 and 2007, and Landsat 8 only image for 2014 and 2021 with a spatial resolution of 30 meters. The processing and the filtering of the satellite images was done on Google Earth Engine environment. And to ground truth, we went to the study area and we collected the ground truth data using GPS for validation and also previous land use land cover images or maps from SANBI for further validation of the classification. This is the flowchart of the method that was used. As I mentioned, we used the Google Earth Engine platform, and the classifier that was used to train the samples was a random forest classifier for the images of 2000, 2007, 2014, and 2021. This is the map of the classified images. As I've already mentioned, that we classified the image for 2000, 2007, 2014, and 2021. And as we can see in these images, the most notable change that happened from 2000 and 2007 is the increase in cultivated land, which is in orange, and there's a little bit of purple there, which is built up. It's not really visible because of the other colors. And we see the decrease in forest land. And from 2000 to 2014, the most notable change was the fact that forest land had been mostly degraded or deforested, because that was one of the land use in the area. They deforested their land to create space for cultivation and also for built-up areas. In 2021, there was completely no cultivated land as the land were abandoned because of lack of water and because of climate change. The classification accuracy is what we see here on the table. And as we can see, the validation overall accuracy of the classification yielded high results, meaning that the results are reliable. As I've already mentioned, this is the post-classification change detection that we were able to arrive at because of the classification that we did in Google Earth Engine, and this was achieved in ArcGIS. As we can see, this post-classification change detection, it shows how land cover changed from 2000 to 2007, from 2007 to 2014, from 2014 to 2021. And as we can see that the most notable color in 2021 is the purple color, which is built up area, meaning that there is a lot of development happening there where many people are moving into the area and they're clearing the land for development. And also there's a lot of degraded area, which is in black, if you can clearly see on the map there. And this is because of those unsustainable agricultural practices that I mentioned, and the fact that the area is really dry and receives really little rain annually. Advanced data analytics and remote sensing application has allowed fast monitoring of land use and land cover changes in Mpanama village. The implementation of Google Earth Engine has been effective for analyzing big data effectively. This overall accuracy achievement indicated that the classification results were both rational and dependable, enabling subsequent post-classification change detection comparisons. The high overall accuracy achieved can be implemented in the random forest classifier, which effectively minimizes errors as the number of decision trees increases. The findings of the study were similar to those obtained by Mashala et al. 2023, where the increase in built-up was attributed to the growing population and the decrease of water bodies due to climate change, while the increase in bare land was due to overgrazing by livestock. The encroachment of bush poses a challenge in Mpanama village, as a study by Lambin et al. 2003 also mentioned, where the invasive species fights for resources such as space and water with the indigenous plants. With the right set of variables, the Google Earth Engine's random forest classifier was accurately used to map and predict the extent and the rate of land use and land cover dynamics. The findings revealed that Mpanama village experienced significant land use and land cover changes between the years 2000 and 2021. During these years, the water bodies dried up, built up, and bare land significantly increased, and bush started to encroach the land. With the primary use of land degradation in Panama being identified, with the primary cause of land degradation in Panama being identified, the GF7 can effectively execute its plan to introduce fencing in order to manage grazing, planting of grass, among others, to achieve land degradation neutrality by 2030. The study recommends the constant monitoring of changes in land use and land cover in the study area, and to hire different scientists to assess the land in order to for appropriate measures to be taken to protect and to restore natural resources in Mpanama village. I would like to acknowledge my supervisors, Prof. Idaw and Prof. KK Aisi, for the support and guidance, the University of Limpopo for allowing me to conduct this research, the Risk and Vulnerability Science Center for the support and for technical data collection issues. I would like to also thank AgriCita and NRF for the sponsor. These are the references that were used to compile this presentation. Thank you.
Thank you, Ms. Mukwebo, for your presentation. The presentation is now open for questions, comments, suggestions. Do we have any questions for Ms. Mukwebo? I see one hand over there. 2, 3.
Thank you. Thank you. Oh, okay. I just want to ask, ma'am, what are the weaknesses of your study, and how can you justify its originality?
Sorry. Hi, thank you for the wonderful presentation. My question is like a general question, it's nothing complicated. You probably mentioned it and forgot. Okay, cool. So the first question is, what motivated the period 2000 to 2021? And in terms of selecting your Landsat images, what criteria did you use to say, okay, I'm gonna use this specific image for this specific month? Thank you.
You can go ahead, Doc.
Sure. Thanks, Chiliti. I just want to pick up your brains on how realistic you think your recommendations are, to reverse land degradation and meet, the land degradation neutrality target. You mentioned fencing some areas. Most likely, some areas are overgrazed, among other things, which led to encroachment. Other than that, these are nice recommendations. How feasible are these?
Thank you so much for the questions. Let me start with the last question. How realistic are my recommendations for the study? Truly speaking, I would say 60% feasible, because this GF7 project started this year, but looking at the fact that 2030 is around the corner, it can take longer to achieve the goals that we want to achieve with the fencing and mitigating land use, land degradation. So we can get there, but at the time limit, I don't think we can make it to the goals that we want, but we can try something in the time space that we have been given till 2030. And what motivated me to choose the years 2000 to 2021? I chose the year 2000 because I needed a strong starting point. The year 2000 is when we experienced floods here in South Africa from Mozambique due to the cyclone. And in that year, that is when we saw a lot of growth in vegetation and also a lot of thriving in forest areas. But looking at the years going further, as shown in the maps, we can see that that amount of water or moisture that the land received was stored in groundwater and also in plant leaves for that period. As we saw that from there onwards, as climate continued to change and the conditions became worse, then we saw that they started to degrade. And also with the land uses, then there were some of the land covers were completely replaced by others. And also I looked at how I can, because I wanted to monitor land use and land cover before the project starts, to see what we will be working with, which now motivated the 2021 as the end year. And from there, I divided the years equally to 7-year periods. From Landsat 5 and Landsat 8, that motivation is because the data for 2000 is covered in Landsat 5, and the Landsat 8 also covers for the year 2014 and 2021. And I wanted also satellite images that would result in accurate results or results that can be useful. So Landsat 8 only came about that way. What were the weaknesses of my study and to justify its originality? The weaknesses of my study, I would say that personally, from what I'm doing, I would say that the weakness of my study, if we looked at the overall accuracy there, I would say that the weakness is only the years 2000. In the accuracy, it was a bit low because the Landsat 5 it had, the resolution there was not really clear in Google Earth Engine. So I would say that was just the weakness there that I experienced. And other than that, there are no weaknesses in my study. Thank you.
Thank you. I think we are running out of time for her. You can still engage with her outside. And thank you for the presentation. Colleagues, we still have one more presenter, Walter Mahaule. please approach the podium. This is our last presenter. He's going to talk about bull fertility in beef cattle.
Good afternoon, everyone. My name is Mahaole ASW, and I'm under the supervision of Dr. Mugwabana, Dr. Nemutandani, and Prof. Diaz. And I'll be presenting my research with the topic factors affecting fertility of beef bulls in South Africa, and it's a review. This is the presentation outline I will be following. Bulls have a major impact on beef production since they contribute to half of the genetic makeup of herds in each subsequent generation. Beef production helps in the improvement of food security in South Africa. Furthermore, beef cattle contribute at least 80% of the total beef herds in South Africa. However, the calving rates of South African communal farmers are lower as compared to those of the commercial farmers. The calving rate of the communal farmers are between 20 and 40%, whereas in the commercial ones they are above 70%. According to these authors, bull fertility is most probable cause of low calving rates. Therefore, understanding the factors which affect fertility in bulls can help farmers in improving the calving rate. There are several studies on factors which affecting fertility of bulls. This author outlined skeletal circumference, type, nutrition, age, and some factors as factors influencing fertility. According to this author, there was positive correlation between skeletal circumference, sperm count, age at puberty, and fertility. However, there are limited studies. There is limited comprehensive literature on factors affecting fertility of bulls in South Africa. The aim of the study was to document the factors affecting fertility in bulls, and the objective was to provide information on factors that affect fertility in bulls. In terms of the study design, a review of data from literature investigating factors affecting fertility of bulls was used, and reports of studies that focused on factors influencing fertility were reviewed. Google Scholar, Science Direct, Web of Science, PubMed, and ResearchGate were the research databases that were used to collect the data. Only English papers were included in this study, and the papers were published between 2000 and 2022 in order to make sure that they are still relevant for the study. And the reviews were excluded, and also duplicates which appeared in more than one search engine. The current study found that skeletal circumference, semen quality management, and age were the major factors affecting bull fertility. Furthermore, skeletal circumference was found to be closely associated with the quality of semen, hence it was the best predictor for fertility. Bulls were considered to have reached puberty when they have a minimum skeletal circumference of 27.9 centimeter, which is normally attained at around 11 months of age. This figure here shows the skeletal circumference and how it's measured. A bull has a minimum of 50 spermatozoa with minimum motility of 10% at puberty, and this increases with age. Furthermore, poor selection and management practices used by smallholder farmers inhibit the improvement of fertility in bulls. And the bulls in extensive farming conditions were found to have a skeletal circumference which is lower compared to those in the intensive farming conditions. And skeletal circumference was found to be best predictor of puberty than age and body weight in bulls of all breeds. In conclusion, it was noted that most studies were assessing bull fertility based on the skeletal circumference due to its relationship with the other factors influencing fertility. This study suggests that the Department of Agriculture should develop programs which are aimed at improving fertility problems in the rural areas based on the identified factors. Furthermore, farmers can use skeletal circumference as prediction method for fertility due to its cost effectiveness and since it's cheap to use, since the farmer will only need a measuring tape to estimate to measure the skeletal circumference. I would like to acknowledge the University of Limpopo and my supervisors for their contribution and also the TIAS research team. These are some of the references that were used in the study. Thank you.
Thank you, Walter, for the presentation. The presentation is open for questions. I can only see two, 1 and 2. Don't you have another one? He is ready to tackle everything that you're going to ask. Okay, you can go ahead.
Thanks, Walter. I have not seen anything relating to, extreme temperature. So, I mean, climate change is one of the contributing measures. I wonder whether if that did not come out in some of the literature you looked at. And maybe just a comment in terms of the breeds, because you just mentioned, bulls. Are all these factors the same across the various breeds?
Thank you. Abigail?
Thank you for your presentation. Well, what I have is not questions, rather comments. Just to add on what was said before, nutrition is also a major factor in this, and I did not see that it was mentioned, so something to look at. And on analyzing the data, I would suggest that you use meta-analysis because you're using other people's data. So that will actually give you a concise evidence across all the publications that you looked at as to which one is the most important factor. Thank you.
Thanks for the comment and the questions. With the question of is the temperature and environment affecting the bull fertility? Yes, they do affect bull fertility and they cause variations in the fertility levels of bulls. And then with the one for if the factors are they the same for all the breeds. Yes, the factors they are the same in all the breeds, but some breeds have different adaptability levels to some of these factors, so they are not influenced necessarily the same. Thank you.
As a follow-up question to the question that was asked, you were asked about if these factors are affecting all the breeds the same. Is it really true that the factors affecting the breeds entirely the same?
Okay.
Now, I was saying, let's rather turn it into a comment because I can see he's a bit unsettled. But if you want to comment, you may. But if not, you may. Leave it. The aim is just to, you know.
Okay. I would say the factors they may affect the bulls differently based on which other factors influence the fertility in the bulls. You may find that the bulls they are not experiencing same factors. Maybe the ones, as I indicated, that the ones in extensive conditions and the ones in intensive, they have different fertility levels, which is because of they are not experiencing same factors such as nutrition and even the lacks of temperature.
Thank you, Walter, for the responses. I think you can be excused now. Please, let's give a round of applause to him. Colleagues, I would like to, I cannot even call him upon on the stage. He's already coming here and he's going to make some announcements. Yes. Colleagues, it has been a wonderful time spending 3 days here. This is the 3rd day. We managed also to be hosting our guests from other institutions across the country. We were having some plenary sessions with our educators. We really thank you. And the members of staff for your strenuous work preparing the presentations for the studying persons. We are doing our PhDs and our masters and our honor students. It's really a pleasure. We could see that our PhD students and our honor students, they are really eager and they are in a transformative way of saying that I am a graduate. So we would also want to encourage you guys to continue to pursue your dreams, to develop your career more, because this is just the beginning for some. And some have been presenting year in, year out. It's okay, and you could see from the confidence that they were exuding in front of the audience here. And those that were doing it for the first time, next time you're going to improve as well. From this moment now, I'm told that the lunch is ready. And I'm also told the venue for the lunch is the usual venue that we were just going. Then there is the gala dinner. It has been scheduled for 5 p.m. today. Remember, we are going for the long weekend. Please, let's stick to time so that we can wrap up this research day conference, and we will have a good weekend. So, ladies and gentlemen, shall we applaud ourselves for the presentations? See you next year.
Okay, the last one, the rooms are ready for changing if you want to change. You will get the key from Professor Masuku. There are 2 rooms for guys and 2 rooms for ladies. Thanks a lot.